Hello. Zoom window screen is not visible.
Hello. Sami, Palak. Good afternoon, sir. Good
sir. Good afternoon, good afternoon. Just now I see the window.
Good afternoon, madam.
Madam?
Hello.
I can't start window. Yes, I can start video. Video is muted.
Sir, good afternoon. How are you, sir?
Good afternoon, sir. Good afternoon, sir.
Good afternoon.
मॅम निलेश जाधवाचा कॅमेरा ऑन करा म्हणून सर. निलेश जाधव सरांचा कॅमेरा ऑन करा म्हणून. हा डॉक्टर.
Hello.
Hi, good afternoon.
Hello.
Hello, good evening, madam. Gladys, madam, professor. Hello.
Hi, can you hear me?
Yeah.
Hello, finally we meet. Thank you very much.
Yeah. Madam, what is the time in Philippines?
It's 5 o'clock. It's 5:08 p.m.
So it is evening now?
Almost 1.5 hours advanced.
है ना? ओके.
नाही नको पुन्हा नको.
Hello, madam. Gladys, madam.
Yes.
Can you hear me?
Yes, I can hear you. Can you hear me?
So,
I'm using a headset now.
Okay, we are going to start at 3 o'clock, but before, after starting, some inauguration program is there, okay? Short.
Yes.
And then we will start, okay?
So I just share screen, right? And then present the talk.
Yes.
Okay.
Yeah.
Yeah. Yes, no problem. Thank you.
हेलो, हाँ बोल ना. How? How? हो ना, ठीक है ना. हो. ठीक.
Hello? Can you hear me?
ते चाललं का मला जात आहे वर्ड फाईल
याला काय करा, याला आपलं पुढे समजा ना, आपल्याला त्याच्यानंतर पाहायचं नाही, आपल्याला फक्त व्ह्यू आणि हो. हो नाही, मी आहे चालू झाल्यावर started with business company. The technology market.
युट्यूबची लिंक आहे ना, पाठवून दे ना त्या तुझ्या स्टुडंटच्या ग्रुपवरती सगळ्यांना. हा ठीक आहे, ठीक ठीक. तुमच्या टीचरच्या ग्रुपवर टाकून.
Hello.
नहीं बिल्कुल
Hello, Chetna. How are you? Chetna.
गुड आफ्टरनून सर।
गुड आफ्टरनून। कैसे हैं?
ठीक ठीक सर।
Hello, Bodke madam.
Good morning, sir.
Good morning. You are ready?
Good morning, sir. Yes, sir.
And where is a Bhaiva sir join नहीं हुए क्या?
Bhairav sir also joined.
Sir. Okay. Sir. Yes. Good afternoon.
How are you, sir?
Yes, fine, sir. Madam has joined. Professor Gladys madam has joined. Okay. Hello, madam.
Yes, sir.
Professor Gladys madam. Here is the Dr.
Hello, good
Bhaiva.
afternoon.
He is the principal of
Hi.
that Guru Nanak College. Yeah.
Good afternoon.
Good afternoon.
And here is a Dr. Josna Khobragade. He is a convener.
Good afternoon, sir.
Wait, I'll do gallery view so that I can see people, maybe.
We can start within 5 minutes, madam. Okay.
Okay, yeah, no problem.
Around three. Okay.
Mm-hmm.
Participants are joining now. And for your
Yes.
information, madam, more than near about 4,000 people have registered
Oh,
for this
oh
webinar.
my
Yeah.
gosh.
Very good
That's a
response is
lot.
there. Yeah.
So is there a live streaming?
Yeah.
Is there a
There is a live
streaming?
streaming on the YouTube. Yes, going on.
Oh, that's why. Okay, now I understand. That's why very few people are here in the Zoom meeting.
Live streaming is going on, madam. Yeah.
Okay, I understand.
Hello,
Thank
Professor Ajit sir. Hello, Professor Ajit. Hello. Hello, Professor Ajit.
Yes, hello, Prof. Do you hear me?
Yeah. You're audible, sir. I'm
Yes, I'm
introducing.
here.
Sir, start your camera. I'm introducing the Professor Gladys from the Philippines.
Is my camera on?
No, it is not on. Make your camera on. Yes, sir.
Is this on?
No. There is an option. See that at the
Okay.
bottom.
Okay, just a second, Prof.
Yeah.
Can you see me now, Prof, or not yet?
No, your camera is off, sir. Make it on.
Is this on now or still off?
No.
On. It's on now. I can see
Okay.
him. Yes. Hi.
Yeah. Yes, sir. Yes.
Good afternoon.
Professor Gladys, madam. He is a Professor Ajit from University of Malaya, Malaysia.
Kuala Lumpur.
Hi. Nice to meet you, Professor.
Nice meeting you.
Professor Ajit, madam is going to give the talk today, and madam is a professor from the Philippines University. Yeah.
All right.
And nice to see you, sir. After a long time, we are meeting.
I think last year February.
In last February.
Yes.
Now this March. 1 year. After 1 year, we are.
After more than a year. Yeah. Just more than a year.
Yeah.
How are the cases in there? The COVID cases, is it going down?
In Kuala Lumpur?
Yes.
I think yesterday about 1,200 new cases. I think coming down for the past 2-3 weeks. It's more than 2,000. More than 2,000 really. Now it's coming down.
Okay. So vaccination already started in your country?
Already started. Just started for frontliners.
Oh, nice. Same with us. And how about there in India, Professor? How do you pronounce your name? Wasudeo?
Yeah, madam. In India also vaccination started every place in India, and also now there is a less number of cases now.
Yes.
But still
That's
some
good.
area having some hotspot.
Yes, same
In
with
some
us.
area, like in Maharashtra state, particularly as compared to other state, there are some good, very big number of the are there patients. About that is the more than thousands, and totally in Maharashtra the cases are more as compared to other state.
Okay.
Hello, Baiva sir. When we will start? Professor Ajij. Professor Ajij.
Yes, Prof.
He is the Baiva. Dr. Baiva is the principal of the college.
All right. Okay.
Baiva sir. Professor Ajij Hassan from the University of Malaya, Malaysia. Baiva sir, now 3 o'clock. Can we start?
Yes, sir.
Okay, Bodke madam.
Should we start, sir?
Yes, madam.
Thank you, sir. Very good afternoon and good morning to one and all for joining today's webinar. Myself, Dr. Yogita Borkhe, Assistant Professor, Guru Nanak College of Science, Ballapur. I heartily welcome all of you for this one-day international webinar on multidisciplinary research IWMR 21, celebrating on the occasion of Golden Jubilee year. With the kind support and immense motivation from Sardar Nagendra Singh Ji Soni Sahib, President Guru Nanak Seva Samiti Virur, we organize this beautiful event. We felt honored to listen to exceptional and extreme personality around the different corner of the world. First speaker is Professor Gladys Completo, Institute of Chemistry, University of Philippines, Los Banos. Dr. Tofida Mohammad Tabir, Federal College of Education, Nigeria. Third is Professor Wasudeo Gurnule, Kamla Nehru Mahavidyalaya, RTM Nagpur University, Nagpur. Friends, unsuccessful people make a decision based on their current situation. Successful people make a decision based on where they want to be. A wonderful lady is none other than the convener of today's webinar, Dr. Joshna Khobragade. So may I invite Dr. Khobragade ma'am for the IWMR remark. So please ma'am.
Am I audible, sir?
Yeah.
Am I audible, sir?
Yes. Hello.
Thank you, sir. A warm welcome to each one of you. On behalf of organizing committee, I welcome all the resource persons, academicians, and participants on this auspicious occasion of international webinar on multidisciplinary research field, organized by Department of Chemistry, Guru Nanak College of Science, Ballapur. First and foremost, one name deserves a special mention, Honorable Sardar Nagendra Singh Ji Soni, who he could not join us but has conveyed his blessing and best wishes for this webinar. Dr. B.M. Mahidwar sir, principal of this college, Professor Gladys Completo from Philippines, Dr. Tofida Mohammed Kabir from Nigeria, and Professor W.B. Gurnule from Nagpur have spared their valuable time to be with us. We all are waiting to hear you all. Professor P.B. Gude sir, coordinator of this webinar, all members of organizing committee, and dear participants across the globe. Multidisciplinary research takes place when a faculty from different disciplines work independently on a common problem or research questions. In this approach, faculty share the research goals and work on the same problem, but look at it from their own discipline's perspective. The advantage to multidisciplinary research is that each aspect can be analyzed by particular specialty, which is often necessary to answer complex research problems. Multidisciplinary approach is very much trained in developing countries to solve the research problem. The main purpose of organizing the international webinar is to provide a platform for interaction and collaboration amongst the eminent professionals researchers, and scholars of the different fields from both academia and industry to discuss and to share the state of the art development. In multidisciplinary research, this webinar will be definitely beneficial to the participant in their field in a general and young student in particular. For this national webinar, more than 4,000 participants have registered from different countries like Thailand, Indonesia, Bangladesh, Philippines, Nigeria, Nepal, Malaysia, Sri Lanka, Pakistan, Libya, Tunisia, Saudi Arabia, Iraq, Afghanistan, UAE, Bahrain, South Korea, Republic of South Africa, Egypt, Mexico, China, Yemen, Ethiopia, Algeria, Canada, and almost all the state of India. This year, Guru Nanak College of Science is now completing 50 glorious years of its service to the nation. And therefore, we have decided to organize the different activities for scientific reasoning attitude among the students and teachers in different diverse areas. We are being blessed and have opportunity to be benefited by the talk of Professor Gladys Completo, University of Philippines, Los Banos, Philippines. Dr. Tofida Mohammad Kabi, Institute Federal College of Education, Jharia, Kaduna State, Nigeria. Professor W.B. Gurnule, Kamla Nehru College, Nagpur, India. Before concluding, as a convener of today's international webinar, I welcome all the resource persons, academicians, and participants. Thank you.
Thank you so much, ma'am. No goal is too high if we climb with care and confidence. With such a wonderful talk, may I welcome the coordinator of today's webinar, Professor P.B. Ghode, Head of the Department, Guru Nanak College of Science, for the departmental remark. Please, sir.
Thank you, madam. Good morning, everybody. Myself, Professor P.B. Ghode, Coordinator and Head of the Department, Department of Chemistry, Guru Nanak Science College, Balharpur. As a coordinator and head, I heartily welcome all the invited speakers, Professor Gladys Completo, Madam, Dr. Tofida, Mohammad, Kabir, sir, and Professor W.B. Gurnule, sir. I also like to welcome our President of Guru Nanak Seva Samiti, Virur, and Principal of our college, Dr. B.M. Bahirwar, sir, and all participants. I am very happy that to have you all with us for this event. Now, on the occasion of the Golden Jubilee of our college, we are organized this international webinar on multidisciplinary research. As you all know that the multidisciplinary research become the part of the today's life, and it is very essential for the development of the mankind, as well as it is very essential for the growth of the society. Now, as a head, it is my duty to introduce our departments. Respected sir and madams, our founder, Ajit Singh Ji Soni, dreamed about the educated society around him, as there was no any college in this region, and hence he has started this single faculty science college in the tribal regions of the Chandrapur district of Maharashtra state. I am very happy that I have the part of this particular college. Dear sir, this particular college, it is established in 1971, and till this date, this college is single faculty college. Now our college, we have one PG department of chemistry, and in PG department of the chemistry, total we have the 7 faculty members, out of which the 5 faculty members, they have the PhD degree, and they are actively involved in their research. Now, in order to enhance the research activity in the college premises and to give the environment to the students and the other faculties of the college. Now, this year, we are going to put the proposal for the CHLR, that is the Center of Higher Learning and the Research. This is the brief introductions about the college. Now, without taking too much time, I would like to express my sincere thanks to all invited guests and for their for accepting our invitations. And I also like to express my sincere thanks to Gurnule sir for encouraging us to organize this particular event in our college. I also express my sincere thanks to our president of Guru Nanak Seva Samiti, Virur, and the principal of our college, Dr. B.M. Bhairavar sir, for their strong support. I really appreciate the efforts taken by the convener of our today's webinar, Dr. J.V. Khogagade madam, to success this particular event. Last but not least, I also express my sincere thanks to anchor Dr. Y.G. Bodke madam. Similarly, our faculty is also involved in all these activities. So it's my duty to express my sincere thanks about our faculty, Dr. Deshmukh sir, Dr. Yadav sir, Dr. Manisha madam, Professor Geda madam, and all non-teaching staff of the college. Thank you very much. And thank you for listening me very nicely. Over to Dr. Bodke.
Thank you so much, sir. Thank you so much, sir. We are very grateful to our honorable principal, Dr. B.M. Bhairavar, sir. Sir, for the encouragement and support. Sir, always help us to reach our milestone. So I request the president of today's functions and principal of our college, Dr. B.M. Bhairavar, sir, for the presidential address. Please, sir.
Okay.
एक आदमी बुखार आ रहा है मेरे को, सबको नहीं आ रहा, सबको नहीं आ रहा.
Good afternoon, one and all. I bow my head to Mata Saraswati, Guru Nanak Dev Ji, and our beloved Papaji, late Sardar Ajit Singh Ji, the founder president of Guru Nanak Seva Samiti, and our Guru Nanak College of Science, Ballarpur, who had planted a sapling 50 years before that now became a huge banyan tree. During this year, we are celebrating Golden Jubilee of our institute. On the behalf of Guru Nanak College of Science, Ballarpur, on this occasion of inaugural function of one-day international webinar on multidisciplinary research organized at Guru Nanak College of Science, Ballarpur, the dignitaries, Honorable Sardar Naginder Singh Ji Soni, Chairman, Guru Nanak Seva Samiti, Professor P.P. Ghode, Coordinator and Head of the Department Chemistry, Dr. J.V. Khobragade, Convener of this webinar, Dr. Y.G. Bodke, Master of Ceremony of this webinar, all faculty members from the Department of Chemistry and other departments. I, Dr. B.M. Bhairavar, Principal of this college, extend a warm welcome to all respected guests, research scholars, participants, and faculty members. The research persons for this webinar are Professor Gladys, Completo, University of Philippines, Los Banos, Laguna, Philippines. Dr. Tofida Muhammad Kabir, Federal College of Education, Zaria, Kaduna State, Nigeria. Dr. W.B. Gurnule, Kamla Nehru Mahavidyalaya, RTM Nagpur University, Nagpur. All scholars, faculties and students across the different colleges in India and abroad. Once again, I welcome you all with my words. Friends, we all are facing the challenges due to the COVID-19 pandemic since the month of March 2020. Since last few months, government started Mission Unlock with certain guidelines to be followed. Recently, government opened the schools and colleges too, and still we are facing the challenges of COVID-19. To some extent, life is slowly becoming normal. Being the responsible citizens, we have to follow the norms issued by government and ICMR to remain away from COVID-19. On this occasion, I take opportunity to introduce our college to international fraternity. The college is creating world-class human resources in the form of scientists, administrators, managers, doctors, engineers, teachers, and the skill hand of almost all fields. Guru Nanak College of Science, Ballarpur was previously affiliated to RTM Nagpur University, Nagpur. From 2012, it is affiliated to Gondwana University, Gadchiroli. Under the dynamic leadership of Honorable Sardar Nagendra Soni, Chairman Guru Nanak Seva Samiti, this college is continued to attain new heights. Our college is accredited B by NAC. Our college offers graduation in basic science subjects and various advanced subjects such as geology, microbiology, biochemistry, biotechnology, and computer science. We have PG department of chemistry. Our teaching faculty is very highly qualified, and almost all faculty members are having doctorate, except very few who are pursuing their doctorate degree. I must proudly inform you to all, every year our students are there in the merit list of university. This year, there are 4 merit students among 10 of the merit list of Gondwana University, Gadchiroli. College is also having collaboration with the various colleges in the university and other universities. To fulfill academic thirst of faculties, we have organized this webinar for faculty members and the research scholars. Pandemic situation provides us opportunities to attend online webinars sitting at home. We should take this situation as an opportunity to update ourselves. Today's international webinar on multidisciplinary research includes 3 disciplines: chemistry, mathematics, and nanotechnology. Professor Gladys Completo is going to deliberate on role of glycans in tuberculosis and human milk, which deals every burning issue, as glycans plays a critical role in host pathogen interactions, antigenicity, and virulence determination, and considered as a potential drug target. Despite of effort taken by WHO to reduce the incident rate, the prevalence of tuberculosis is increasing in certain regions. This talk by Professor Gladys Completo may give review insight into the role of glycans in mycobacterium pathogenesis and its implication in drug development. Dr. Tofida Mohammad will be delivering a talk on effect of viscous dissipation on steady natural convection covert flow in vertical plates. Dr. W.B. Gurnule will speak on nanotechnology and its impact on society, as nanotechnology will have significant social impact in the various fields such as military applications, intellectual property issue, as well as having effect on labor and the balance between citizens and the government. Dr. Gurnule will throw more light on impact of nanotechnology on society, which will definitely beneficial to researchers in this field. Scientific progress relies on problem solving and collaboration. Group composed of people with diverse experiences and areas of expertise tend to be more creative and innovative. More than 4,000 participants have been registered for this international webinar on multidisciplinary research, which is very, very creditable for us. I hope participants will be definitely benefited by this webinar. Wishing great success for this international webinar.
Thank you very much all of you.
Thank you so much, sir, for your word of wisdom. It will be a great pleasure for us to listen you again in the future events. Participant, now we are entering in the first technical session, which is based on the glycans in tuberculosis and human beings. So I am inviting Ms. Sonal Geda, Assistant Professor, Guru Nanak College of Science, for the brief introduction of our speaker one. Please, ma'am.
Very warm good morning to one of each. I am Sonal Geda, Assistant Professor from the Chemistry Department. On behalf of Guru Nanak College of Science, Balapur, I heartily welcome all the dignitaries, faculties, and all the participants on the occasion of international webinar. It is my absolute honor to introduce our first speaker, Dr. Gladys Completo ma'am. Dr. Gladys Completo ma'am completed PhD in Organic Chemistry at the University of Alberta under the guidance of Dr. Toj Laris, where she was trained as a synthetic carbohydrate chemist. It is very proud to share that Madam has awarded 2 postdoctorate fellowships. She subsequently did postdoctorate fellowship in the glycobiology with the Professor James Paulson at the Swift Research Institute. This was followed by another postdoctorate fellowship in the medicinal chemistry at the University of California, Los Angeles with the Professor Michael Jansen. Her main area of research involved in the molecular biology of a disease such as tuberculosis and cancer. In the field of research, ma'am also visiting research professor in Professor Canito Labrila's lab at the University of California, Davis, USA, training on mass spectrometry based on the clinical glycomas. She is currently working on collaborative research such as biomarker discovery for the lung cancer using mass spectrometry based glycoproteomics. Besides teaching and research, she is actively involved in various professional organizations such as Chemical Society of Philippines (ST), where she served as president in 2017, and also in Philippines American Academy of Science and Engineering as the Chemistry Research Cluster Chair. Now, Dr. Gladys Completo ma'am is currently working as professor at the University of Philippines, Los Banos. It is very proud moment for us. Such a young intellect and the epitome of knowledge is with us on this occasion. Now I request Dr. Gladys Completo ma'am to deliver their lecture. Thank you so much.
Thank you, ma'am. May I request the speaker one, ma'am, to please start your session. Are you ready, ma'am?
Yes. Thank you very much for that very kind introduction. So I would like to share my
Gladys
presentation
Completo.
now. Yes. Can you hear me?
Yeah, madam.
Yes. Okay, good afternoon, everyone. Again, thank you for that kind introduction, and I'd like to thank the organizers for inviting me to be one of the speakers in this international webinar on multidisciplinary research, IWMR 2021. Am I muted? I'm not.
No. Yes. Madam, go on. Okay, it's okay. Audible. You are audible.
Okay. And then, so yes, I'm honored to be part of the Golden Jubilee Year celebration of your college. So I'd like to talk about the role of glycans in tuberculosis, and I was supposed to give a talk on also glycans, the role of glycans in cancer, but I decided to talk about my other project, which is on human milk oligosaccharides, and hopefully I would be able to attract some collaborators on this project. Okay, so let me start now. 10 million. This number corresponds to the number of people that got infected with tuberculosis in the year 2019 and 2020. So statistics have shown that 1/3 of the world's population is infected with TB. In fact, they said that every second, one person gets infected with this disease. And the reason why it's so difficult to treat tuberculosis is because of the emergence of what you call multi-drug resistant strains, or MDR-TB. And recently, the emergence of what you call extremely drug resistant TB strain called XDR-TB. So the World Health Organization was alarmed, and they came up with this sentence that XDR-TB is basically a death sentence. And they created a program which they called the NTB strategy, with a goal that by year 2035, there will be 0 deaths caused by tuberculosis. Okay, aside from the emergence of the MDR-TB and XDR-TB, another reason why tuberculosis is so difficult to treat is because of the highly impermeable nature of the mycobacterial cell wall. This is the caricature representation of the mycobacterial cell wall. And the moiety responsible for the impermeability of Mycobacterium tuberculosis is the mycolyl AG complex, comprised of mycolic acids, arabinon, and galactan chain. We're particularly interested in the galactan chain of this complex because it is responsible for the survival of the microorganism. And also, it is composed of galactose residues, which are in the form of furanose, which is not present in human beings. Okay, so this is the more detailed structure of the AG chain of the mycoarabino-galactan complex. As you can see here, it is built on the core of galactan chain, which is composed of 31 galactose residues. As I mentioned, it's not in the usual form, which is the pyranose form of a sugar, but it's in its 5-membered form, furanose ring. What's more interesting about this galactan chain is that it is composed of alternating beta 1,5. So this one, the first position, the fifth position, this beta 1,5 and beta 1,6. And it's alternating throughout the galactan chain. Okay. Now, the biosynthetic pathway for the synthesis of this complex has been proposed. Okay. It involves the sequential addition of sugar residues that starts with glucnac, followed by rhamnose, and followed by the galactofuranosides before the addition of the arabinofuranosides. This polyprenyl bound intermediate then is transferred to peptidoglycan before it is added to the mycolic acids. So very little is known about the glycosyl transferases involved in the formation of the mycolyl AG complex. But as I've mentioned earlier, we're just interested in these galactosyl transferases. During the time when we started this research, we thought that there's only one galactosyl transferase involved in the formation of the galactan chain. And another question that you might ask is, are these enzymes involved in the mycolyl AG complex formation viable targets for drug action? In fact, most of the first-line drug that is being used to treat TB, such as isoniazid, is known to inhibit the formation of the mycolic acids. Another drug, first-line drug to treat TB, ethambutol, okay, is known to target the arabinon chain. But none of the known anti-TB drugs is used to target the galactan chain. And since it's been reported to be responsible for the survival of the microorganisms, this will be a very good drug target. And also because it's composed of galactofuranoses, which are not present in human beings, then it will minimize possible side effects. That's why we're very interested in this galactan chain. Okay, as I mentioned earlier, at the start, we thought that there's only one enzyme responsible for the formation of the entire galactan chain, and we call this as Gel Fd. What's interesting about this enzyme is that it is bifunctional. It is responsible for the transfer, for catalyzing the transfer to form the beta 1,5 linkage, and also for the formation of the beta 1,6 linkage. Okay, But we have a theory hypothesis that if we try to examine the linkages present in the galactan chain, maybe there are other glycosyl transferases present. So these are the questions that we want to answer. Can the enzyme Gel Fd synthesize the full galactan chain? Or are there differences in the activity of acceptors of different sizes, as you will see later on? And also, are there differences in the efficiency of 1,5 transfer versus 1,6 transfer? And when we were starting this project, we didn't have the crystal structure of the protein yet. We wanted to know whether there are 1 or 2 active sites because of the bifunctional nature of the enzyme. So this is the entire mycobacterial galactan chain structure. And our hypothesis is that there are 3 different galactofuranosyl transferase involved in the formation of the galactan chain. If you look at the linkages, there are 3 different linkages present. This one, the beta-1,4, that connects the first Gal-F residue to the linker unit. The second galactofuranosyl linkage is this, showing the beta-1,5 linkage. And the third type of linkage is the beta-1,6 linkage. So here's our hypothesis. So maybe there's a first galactosyl transferase, and we can call it as GAL-T1, that is responsible for the transfer of the first Gal-F residue. And maybe there's a second galactosyl transferase we can call GAL-T2 to form the first beta-1,5 linkage, and followed by a GAL-T3 that would form the first beta-1,6 linkage. And then finally, the Gel Fd enzyme that we have in our lab that is responsible for the formation of the full-length galactan chain. Okay, for us to be able to answer this hypothesis, okay, and test the hypothesis, these are the oligosaccharides that we synthesize in order to understand more about the enzyme, where we have disaccharides with either the beta-1,5 or 1,6. We also prepared analogs of the disaccharide where we remove the terminal hydroxymethyl group. And we also have trisaccharides with the alternating beta-1,5, 1,6, or the reverse, and longer oligomers such as the tetrasaccharide with still the same alternating linkages. And in addition to this, we also synthesized these other substrates where we wanted to know what is the effect of the linker unit, the presence of the linker unit. Is there another enzyme that would be responsible in introducing the first Gal-F residue to form this compound? And we also form longer oligomers to test further if there are other galactosyl transferases that might be involved in the formation of the galactan chain. And just to make the story short and to make you excited about it, it turned out that there are only 2 galactosyl transferases involved in the formation of the full galactan chain. And these are what you call Gel Fd2, which is responsible for the introduction of the first and second Gal-F residue. And Gel Fd2 is also bifunctional. And this is followed by Gel Fd2, which was the original Gel Fd in our lab, which is responsible for the formation of the full-length galactan chain. As you can see here, the pink sphere represents the one with the beta-1,5 linkage, and the blue sphere represents the beta-1,6 link Gal-F residues. Okay, when we started synthesizing the galactofuranosides, we started using some of the reported methods that are available in the literature. So we tested Fischer glycosylation, high temperature benzoylation, and even started with this aldono 1,4-lactone precursor. But it turned out that these are very difficult reactions and we always get impurities. So we decided to look up the literature And we found this very simple paper, okay, that was published in the 80s, where they only used iodine and alcohol, and they were able to produce galactofuranosis. Okay, but to our surprise, they didn't use galactose. Okay, they only use glucose, okay, and other monosaccharides except for galactose. Based from this, we came up with our own optimized method, and we were able to synthesize methyl galactofuranoside from this dithioacetal with the use of very simple compounds, methanol and iodine, without any presence of pyranosides, and with very high yield. Because of that, we started using this method in synthesizing our galactofuranosides. We even modified our alcohol, where we use, instead of methanol, we use octanol, so that we can make our A glycan longer to represent the lipophilic A glycan. Okay, so here is the synthetic pathway that we use to synthesize our acceptors, where we started with the dithioacetal, and upon addition of octanol and iodine, we produce, and followed by benzylation, we produce this fully benzoylated octyl galactofuranoside, followed by a series of deprotection and protection. And once we have this fully protected octyl galactofuranoside, we added trifluoroacetic acid, and by the migration of the OH group, we were able to yield two types of acceptors that we needed for our synthesis of the oligosaccharides. So we didn't have to go through different steps. With the acyl migration was actually an advantage for us, because we needed this 6OH position free and the 5OH position also free. And this serve as our acceptors for the synthesis of our longer oligosaccharides. Okay, we use the same method in synthesizing the donors, where we have the dithioacetal, and this time we use the methanol and iodine to form methyl galactofuranoside. This was then reacted with parathiocresol in the presence of a Lewis acid BF3 iterate to form our fully protected thioglycoside, which will serve as our donor. This was followed by the same deprotection and protection steps, and we also made use of the TFA deprotection so that we could achieve the two types of thioglycosides, and this was further acetylated so that later on we can use selective deprotection of an acetyl group in the presence of benzoyl groups for our synthesis of our longer oligosaccharides. And then, so this is just an example of the synthesis of one of our of the disaccharides. You can see here that we're starting with this donor thioglycoside, and this was coupled with the needed either this octyl acceptor or the other octyl acceptor, which was coupled in the presence of NIS silver triflate to form the disaccharides that we needed, followed by Zemplin deacetylation to form disaccharides 1 and 2. We also use this same synthetic method in synthesizing the analogs of the disaccharide, where we remove the terminal hydroxymethyl group to form these two analogs. In the synthesis of the trisaccharides, we decided to use the one-pot synthesis, where we used the trichloroacetamidate as the donor, okay, and this was coupled with a thioglycoside acceptor. Remember, in the previous slides, we used thioglycoside as the donor, but in this case, it can be used as an acceptor with the imidate as the donor. This was activated with DMS triflate to form the desired disaccharide, okay, in situ, because it's one-pot synthesis. this thioglycoside then can now act as the donor, and upon addition of the acceptor with activation of NIS silver triplet, we were able to produce disaccharide in 41% overall yield without doing the very long process of protection and deprotection. So this made our synthesis much shorter and more efficient because we needed to prepare these trisaccharides in large scales to study more about the glycosyl transferases involved in the formation of our galactan chain. Because this was very effective, we also used this method in synthesizing our tetrasaccharides. So we decided to form first the, this is one method where we prepared first the trisaccharide as the donor, and then this was coupled with the monosaccharide acceptor. This thioglycoside trisaccharide can be synthesized from a disaccharide imidate and a monosaccharide thioglycoside. So this is the synthetic route that we use in making our tetrasaccharides now. You can see that this is just very similar to the one-path synthesis, but in this case, we did it sequentially, okay, to form now our thioglycoside trisaccharide donor, which was further coupled with our octyl furanoside acceptor, which gave us our fully protected tetrasaccharide, and followed by deprotection to form our needed tetrasaccharide 8. The same route was used in forming the other tetrasaccharide. So in this case, this is a 1,6 beta 1,6 beta 1,5 beta 1,6 linked tetrasaccharide, and we needed to prepare the other linkage where it started with beta 1,5 followed by 1,6 and then 1,5 linkage to form the reverse tetrasaccharides. Now, once we have all of our oligosaccharides ready, we needed to test these compounds, and we use a radiochemical assay that involves 2 steps. The first step involves the formation of what you call UTP-Gal-F, which serves as the donor in the enzymatic reaction. Okay, and then this is followed by the transfer of UTP-Gal-F, of the Gal-F residue from UTP-Gal-F to the growing galactan chain, catalyzed by the enzyme Gel Fd2, Gel Fd rather, to form our desired product. The radiochemical assay can be traced by using a tritium-labeled Gal-F here at the C6 position. So we tested the products that formed, but once we did the enzymatic reaction, and you can see here that if we started with trisaccharide 1,5,1,6, you can see here this beta 1,5 and beta 1,6, it forms the tetrasaccharide beta 1,6,1,5,1,6 link product. And this was confirmed by the mass spectrum that we obtained, where you can see here that there's still remaining trisaccharide starting material, and where the longer oligomers are formed, not just the tetrasaccharide, but you can see the other longer oligomers with the pentasaccharide, hexasaccharide, and heptasaccharide present. Okay, so this is just one proof that our reaction is actually happening, and then we're getting the products, the longer oligomers. But aside from them, we have to test whether we're getting the right linkages. So we needed to do Nmr characterization for the products. And it turns out that, okay, by just doing the proton Nmr, we noticed that the beta 1,5 link disaccharide, the proton that is attached to the beta, the anomeric proton here, resonates at a more downfield position compared to the reducing proton at the proton at the reducing sugar here, which is 4.96. And when the linkage is beta 1,6, it is a little bit upfield compared to the beta link disaccharide. As you can see here, for the longer oligomers, these still observe. You can see that upon the addition of another Gal F residue with a beta 1,6 linkage, you can see that the chemical shift is 5.04, which is consistent to this disaccharide, followed by the beta 1,5 link, which is also consistent to what we observed with the disaccharide. And this was further observed with the tetrasaccharide that we synthesize. So in general, for the beta 1,5 link residues, we found that the protons resonates at this chemical shift, 5.21 to 5.23 ppm, followed by a more upfield resonation at 5.02 to 5.04. And the proton at the reducing end resonates at the 4.96 to 4.99. But of course, this was also supported by 2D NMR studies. We still did GCOSY, HMQC, and TOXY to verify the structures of our compounds. To just give you an example, there's one of the spectrum that we obtained when we did the enzymatic reaction for this trisaccharides with a 1,5,1,6 linkage. We would expect that the product will be a 1,6,1,5,1,6 link tetrasaccharides. And you can see here that there is only 1,1,5 link proton found at this chemical shift, and 2,1,6 link protons found at the near 5.0 ppm, which just proves that we were able to synthesize the right product. Okay, as I mentioned earlier, our hypothesis was that there are 3 galactophenaracyl linkages present in the galactan chain, so we thought that maybe there are 3 glycosyl transferases involved, right? So we thought that there might be one GAL-T, 2 that is responsible for incorporating this first Gal-F residue, followed by another GAL-T, and the third GAL-T transferase. But it turned out that, okay, this is our hypothesis that I already mentioned before. And in order for us to test this hypothesis, we needed to synthesize more substrates with a linker unit to represent this, the part of that galactan chain. So we prepared more oligosaccharides to test whether there are other glycosyl transferases involved in the formation of the galactan chain. And then these are just the synthetic pathway in synthesizing first the linker unit 9, the disaccharide. Another synthetic pathway in synthesizing our trisaccharide with a linker unit. I won't be elaborating more the details of the synthetic route. This involves it just merely involves protection and coupling, followed by the protection to form the desired trisaccharide. And this is the synthetic route for the formation of the tetrasaccharide that we needed. We then tested this for oligosaccharides with our enzyme, and this is now the synthetic pathway for making the pentasaccharide, okay, by coupling the trisaccharide with the linker unit to form now our last target molecule, which is the pentasaccharide 12. Then we tested this with our enzyme Gel Fd, and it turned out that this linker unit is not recognized by Gel Fd at all. There was no product observed. And we also tested the linker unit with 1 GalF residue, and you can see that there was no reaction. It was only when we tested the tetrasaccharide where there are now 2 GalF residues present with the linker unit when Gel Fd was able to recognize and showed the presence of the product, the pentasaccharide. We then further tested the tetrasaccharide, and you can see that the pentasaccharide was synthesized. And when we tested the pentasaccharide with a linker unit, Gel Fd can still recognize it to form the hexasaccharide. All of these products were confirmed by testing them, by elucidating the structures by NMR and mass spectrometry. So as you can see from the previous slides, that these 2 substrates cannot be recognized by our enzyme Gel Fd. We suggest that there are actually other glycosyl transferases that might be involved in the formation of the galactan chain. And when we were making this, it turned out another research group was actually studying the enzymes involved in the formation of the galactan chain, and they were able to identify a novel galactosyl transferase, which they named as Gel Fd2, which is also bifunctional. When we learned about this, we decided to submit the oligosaccharides that we synthesized to test for them to test it against their enzyme. And these were the results. So you can see here that the disaccharide that was not recognized by our enzyme in our lab was recognized by Gel Fd2 to form the desired product with the right linkage here. And when the trisaccharide was also reacted with Gel Fd2, they were able to see the formation of the desired product tetrasaccharide. But when they further tested the tetrasaccharide oligosaccharide, where you can see here there's a disaccharide now with a beta-1,5 linkage, Gel Fd2 was not able to recognize it, just proving that another enzyme is acting in the formation of the longer oligomers starting from this substrate. So based on the synthesis of all of these oligosaccharides, where you saw that we synthesized the disaccharides and disaccharide analogs and trisaccharides and tetrasaccharides, and the oligosaccharides with the linker units, showing the sequential addition of the GalF residues starting from 1 to 2 and 3 GalF residues, we were able to show that indeed there are more than one glycosyl transferase involved in the formation of the galactan chain. And we were able to, because of that collaboration, we were able to publish another paper with that research group where they tested our synthesized oligosaccharides with their enzyme and were able to establish indeed that there are 2 enzymes, Gel Fd2 and Gel Fd2, which are both bifunctional enzymes. Our original Gel Fd is now called Gel Fd2. So based on these results, okay, we are now doing synthesizing probes to test the specificity of much simpler substrates. These are the work now of my graduate students and undergraduate students, where we wanted to, sorry about this, there should be an oxygen here, okay? We wanted to simplify and come up with potential inhibitors of the Gel Fd2 enzyme. And so based on the synthesized disaccharide that we did before, we wanted to check whether this hydroxyl group at this position, okay, the penultimate residue, is important, or is this hydroxymethyl group in the non-reducing end, is it important for the recognition of Gel Fd2, and possibly we can design inhibitors. So what we did was we synthesized Okay, disaccharide 1, where the hydroxymethyl group was removed here, and disaccharide 2, where both hydroxymethyl groups were removed in both residues. We then proceeded with the synthesis. I won't show you the synthetic route anymore. I'm proud to say that these were synthesized by 2 undergraduate students, and it is very difficult to do synthesis in the Philippines, but they were able to successfully make these 2 compounds. And then we submitted this to the University of Alberta, to my previous PhD advisor, where they did MALDI-TOF MS analysis of the enzymatic reaction mixtures, where it involves the incubation of the potential substrate with the enzyme and the donor, and of course the other enzyme that converts the UDP-Gal-P to UDP-Gal-F. This was then purified using SEPA cartridges, and then the reaction mixture was analyzed using MALDI-TOF MS. And our results show that we even did kinetics of this substrate that we made. In addition to the MALDI-TOF MS analysis, we also did spectrophotometric assay for Gel Fd2, and this was also done in collaboration with Dr. Tad Lowry. And this involves a coupled assay where the compounds being tested are coupled with the oxidation of NADH, which is being observed at 340 nanometer. And the Gel Fd activity is directly proportional to the rate of oxidation of the NADH. And here are the results that we found. Okay, so here's the mass spectrum that we obtained for the disaccharide that we synthesized here, the first disaccharide, where the hydroxymethyl group at the reducing end is absent. And it turned out that Gel Fd2 can recognize this new compound that we synthesized, and even further elongated up to tetrasaccharide longer oligomer. And and a new spectrum was just sent to us that the further longer oligomers were also produced. And this just shows that this hydroxymethyl group here at this position is not that important for it not to be recognized by Gel Fd2. We even did kinetic studies, and we were able to get the Km, Kcat, and the Kcat over Km of the enzyme using this compound as a substrate. So compound 1 is definitely a substrate that can be elongated by Gel Fd2. How about the other compound that we synthesized? Okay, is it also a substrate that can be recognized by Gel Fd2, or is it a potential inhibitor? And the main difference is we remove this addition, the second hydroxymethyl group. And here are the results. Okay, so we were expecting that there will be an addition of the next Gal-F residue, but it turned out that compound 2 is not a substrate, so there's no elongation observed. And we tested whether it can be a potential inhibitor using the coupled spectrophotometric assay. And as you can see here, we have our test substrate, a known substrate that is recognized by Gel Fd2. And you can see that compound 2 is neither a substrate nor an inhibitor. Okay. Although this gave us negative results, this actually helped a lot for us in so that we can design potential inhibitors of our enzyme. We can design better. In addition to that, okay, we decided, okay, to design more compounds and synthesize more compounds because of our finding that this hydroxymethyl group, okay, is not an essential requirement for the beta-1,6 transferase, but this hydroxymethyl group in the terminal Gal F residue is necessary for Gel Fd2 recognition. And during this time, the crystal structure of Gel Fd2 was already available. Okay, sorry about that. There should be a picture here. We designed new analogs where this part of the sugar, okay, sorry, where this part of the sugar is replaced by these moieties. And we also did computational studies on the designed potential inhibitors. This is in collaboration with another professor in our university, where we asked them to do computational studies on our designed potential inhibitors. And they also did 3D QSAR to as other additional design potential inhibitors of Gel Fd2. And we were able to come up with a pharmacophore that is recognized by Gel Fd2. And this is just an example of one of the compounds that we tested that was docked in the Gel Fd2's active site. And you can see here that the aspartate 372 is very important in the hydrogen bonding with either the 5-OH group or 6-OH group of the galactofuranose residue. So here's the pharmacophore that we were able to identify from if we're going to design a donor substrate-based inhibitor. This UTP Gal F residue is important, and the R group here originally is composed of the phosphate group. It has to be charged with the linker increasing, but with the affinity also increasing, but decreasing in permeability. And it turns out that these 5-OH groups here, 5-OH and 6-OH position, is very important. And for the binding to increase, okay, binding towards Gel Fd2 to increase, it needs to get rid of the hydrogen bonding. That's why in the inhibitors that we designed, the presence of methyl group or other moieties which are lipophilic, which doesn't involve hydrogen bonding, resulted to a better binding. And also for the QSAR based design, it turns out that these groups are important, where you can incorporate any lipophilic group, okay? A shorter carbon linker will allow for less flexibility, and the presence of acyl group also increases the binding affinity. And if we wanted to also design an acceptor substrate-based potential inhibitor, again, it's very important that we have a long lipophilic aglycan, and we need to remove one of the hydrogen bonding interaction in either the 5-OH or 6-OH position. And how important is molecular docking in the design of potential inhibitors? It's faster because just based on the computational studies, it can guide us which compounds are more likely to become a better inhibitor. And because of that, we'll be able to discover new drugs for tuberculosis. That is with a new mode of action that is targeting the galactan chain, which is responsible for the survival of the microorganism. And also, I've mentioned before, these galactofuranoses are not present in our body, which minimizes potential side effects, and it will be mycobacterium specific. Okay, so these are the findings that we have done, made so far. And aside from the synthesis of the Gel Fd2 enzyme inhibitors, we are also exploring potential inhibitors of the other enzyme Gel Fd1. Unfortunately, because of the limited time, I won't be able to share our results on that. Now, I would like to switch gears and move on to the next topic that I would like to share. Okay, so I decided to change the topic. Instead of giving a presentation on the role of glycans in cancer, I would like to give my presentation on human milk oligosaccharides that I am working on. And the reason for this is that I'm hoping that I will get collaborators from your university, okay, and even for potential collaborators for the TB project. Well, I started my slide with the number 10 million in my TB presentation. I would like to start again with this number. This time it's 1,000. And this number represents the first 1,000 days of life that starts from the conception up to the second birthday of the child. And this is considered to be very important because it is responsible for the child's growth and development. The World Health Organization has recommended exclusive breastfeeding during the first 6 months of a child. Unfortunately, very few Filipinos practice exclusive breastfeeding. We even spend a lot in purchasing infant formula, and even the low-income families would opt to purchase an infant formula milk. And one of the reasons why they purchase infant formula is because they don't produce enough milk, which is just a sign of that the mother is not healthy. This is a picture of one of the faces of Filipino children with severe wasting. This is the picture of Jomar. And as you can see here, Jomar has a huge head diameter compared to his body size. Okay, and this is a picture of a mother and daughter that are undernourished, and this mother decided to not to breastfeed her child because of this misconception that infant formula give the same benefits as breast milk. So let me just give you a report by UNICEF in 2019, where Philippines is one of the countries in the world with the highest incidence of malnutrition. But aside from the Philippines, there are other 3 regions. You can see this is Philippines, and here's India, where it is considered as one of those countries having highest incidence of malnutrition, with a ratio that 1 out of 3 children below 5 years old is actually stunted. And what's more alarming is that although there is still an increasing number of undernourished children in the Philippines, there is also an increasing number of overweight and obese children, making Philippines suffering with the double burden of malnutrition. So clearly there's a problem. There's an insufficient reinforcement of breastfeeding among Filipino population, and mothers are not knowledgeable on the benefits of breastfeeding. And I don't know if this is happening in India. There's an aggressive campaign from companies where they claim that their infant formula are clinically proven to give the IQ and EQ advantage, which is very enticing to mothers. So in this study, before we collect samples from Filipino volunteers, we conduct seminars on breastfeeding and tell them the benefits of breastfeeding. And I would like to tell this is the first comprehensive study that have shown the human milk oligosaccharide profiles of a lot of Filipino mothers. And I will tell you later what is the importance of this phenotypic secretor status that can be determined by just testing the breast milk of mothers that are breastfeeding. Okay, so what are the components present in breast milk? Okay, you can see that it is mostly composed of lactose, followed by lipids and proteins, and these human milk oligosaccharides, which is more commonly known as HMO. HMOs are comprised of these 5 different monosaccharide units with the core structure lactose moiety, and it is also classified as either fucosylated, sialylated, or non-fucosylated neutral oligosaccharides. What's very interesting about the HMOs is that they're actually not digestible in the infant gut. In other words, it has no nutritional value to infants. In fact, 90% of these are excreted in the feces. So with no nutritional value and 90% being excreted in the feces, what are the functions then of the human milk oligosaccharide? One function that is very important is that it serves as prebiotics for beneficial bacteria, which is responsible for the production of short-chain fatty acids in the infant's gut. Aside from being functioning as a prebiotic, another function of HMOs is that it acts as a soluble decoy. For an infection to happen, the pathogenic microorganism should bind to the surface of the intestinal mucosal cell. But since the structure of the HMOs is very similar to the structure of the glycocalyx, this is the surface of the intestinal mucosal cell, since the structure are very similar, they can serve as soluble decoys, which leaves the pathogenic microorganism attaching to the HMOs instead to the intestinal mucosal cell, thereby inhibiting infection. In other words, if an infant is being fed with the infant formula, there are no HMOs present there, which can lead to infection, while a breastfed infant will most likely have no infection because of the presence of HMOs. So it serves as a prebiotic, it serves as a soluble decoy, and it can also serve as a glycan modifier, where it induces the chains of the structure of the sugar present at the intestinal mucosal cell, thereby inhibiting again the infection of this pathogenic microorganism. And HMOs are known to have sialylated or sialic acids. Okay, and it's known that since brain gangliosides are composed of sialic acid, it has been reported that HMOs are also involved in the brain development of the infant. So these are just some of the examples of the functions of HMOs. And we determine the production of HMOs by genetics. So secretory individuals secretes these ABH antigens in their bodily fluids such as saliva, blood, and breast milk. And a secretor mother would produce a huge amount of this 2'FL, while a non-secretor mother would produce a lot of amount of this LNFP3. And the reason for this difference in the production of the oligosaccharides is due to the presence or absence of the gene responsible, gene Fd2, which is responsible for the introduction of the fucose in this disaccharide moiety. So this is present, Fd2 is present in secretors, while Fd2 is absent in non-secretor mothers. To compensate for that though, non-secretors produce more of the sialylated and neutral oligosaccharides. So our study, this study is being done in collaboration with a professor in UC Davis, where we made use of a high throughput mass spectrometry-based glycomic studies, where we can actually use a 96 well plate and only consume 25 microliter of the breast milk. This is followed by defatting and protein precipitation. And once the oligosaccharide is isolated, it is further reduced and purified before it is introduced into the LCMS instrument. And based on the retention time and the mass charge ratio available, and also the library that's available in their lab, we were able to identify this huge number of oligosaccharides present in the breast milk of most of our samples. And it turned out that 81% of the Filipino donors are secretors and 19% are only non-secretors. We also tested, these are just some examples of the sugar structures that we were able to identify present in breast milk. And we also determined the different oligosaccharides that are present in secretor mothers. And you can see here that the fucosylated oligosaccharides are highly expressed in secretors, while the sialylated and the fucosylated neutral HMOs are highly significant for the non-secretor mothers. Okay, and across lactation stages, you can see that there's a decrease in the oligosaccharide concentration across lactation stages, where the highest amount in the HMO starts from the week 1, where it is in, they produce colostrum, and going down across several lactation stages. So we tested from colostrum to from 1 month colostrum, and then month 1 up to the 6th month of lactation. So right now we're now comparing the milk oligosaccharides present not just in humans but also in animal milk. So if you look at if you try to compare the human milk composition of the human milk and the cow's milk, you can see that they're very similar in the composition of lactose, protein, and fat, but not in the presence of the oligosaccharides. You can see here that the animal milk has 100 to 1,000-fold less in amount in the oligosaccharides compared to the human milk. We also tested the composition of buffalo milk, and we tested different breeds, and we compared it with the bovine milk oligosaccharides and the human milk oligosaccharides. And you can see here that compared to bovine milk, buffalo milk is actually better, since it produces sialylated and fucosylated oligosaccharides, which are present in human milk, but not present in bovine milk. Okay? But also present in all of the buffalo breeds. And the most interesting part is that the sialic acid that is present in buffalo milk is the same as the sialic acid present in human milk. That is the N-acetylnuraminic acid. So it has no new 5-GIC, which is the only sialic acid present in bovine milk. So the reason why we're doing this, because we want to show that buffalo milk can be a better substitute for human milk because of the absence of new 5-GIC and the presence of the sialylated fucosylated oligosaccharides. So let me just show you the advantages of a secretor mom versus a non-secretor mom. So both of them, okay, promotes the growth of, formation of a healthy microbiota because they're both prebiotics. But if an infant is taking milk from a sick-feeder mom, then the infant is less prone to diarrhea and less prone to bacterial infections. But an advantage of an infant taking milk from a non-sick-feeder mom is that it will be less prone to viral infection and even digestive tract cancer. So both have advantages and disadvantages. And our collaborator is actually studying all of these HMOs, and they're collecting milk samples from different countries. And you can see here, this is our result where we use 155 samples. And from India, we only tested 43 samples. So we welcome collaborators who would be able to provide us more Indian milk samples. Okay, and you can see that in most of this population, there are more secretors compared to the non-secretors, except for Bolivia, where it was very surprising that they are all secretors, but with just a small amount of volunteers. Okay, so this is the study that we've been working on, human milk, and this is, I'd like to acknowledge the funding. This is the UP Balik PhD funding that I was able to get for the TB project. And this is the oil funding for our milk project. And this Anthony was the one who worked in UC Davis who came up with all those results. And we call our research group as GlycoBio Research Group. And these are some of my students, and most of them already graduated. And this guy here was the one who did all the computational studies that I've shown you. These are the people that work on the TB project. These are the 2 undergrad students who were, I was very fortunate to have very good undergraduate students. And these are also some of our other additional undergraduate students that work on our TB project. This is a picture of my old lab, and now we have a really nice lab due to the fundings that we were able to obtain. And this is our collaborator, Dr. Carlito Lebrina from UC Davis. And aside from that, we also have medical doctors as collaborators from the Lung Center of the Philippines. And we even collaborate with the industry, the Pascual Pharma. This group is the one working on the other project that we have that is on the biomarker discovery on lung cancer. So these are our graduate students who've been working really hard during the pandemic. They were very productive, and they still practice distancing, as you can see here, and they were masked. And I'd like to share to you our campus, this University of the Philippines, Los Baños. We are located at the foot of a mountain called Mount Maria Makiling Forest. So, and for that, I'd like to thank you for your attention, and I'm welcome to address some questions. Thank you.
Hello, ma'am. Am I audible?
Yes, I can hear you.
Dr. Gladys, it was very informative talk and was full of scientific information. Being from a microbiology background, I have just a curiosity. As we know, there are 3-4 strains of mycobacterium, that is MDR, that is TDR, and XDR. I would like to know, is there any difference in the glycobiology or glycoproteomics of these variants of the mycobacterium?
We actually haven't studied that yet, but yeah, definitely, I'm sure there will be some differences. But maybe in the in the glycan structures at the surface, but since the galactan chains is a little bit in the middle, we're not sure if there will be a difference. Okay. I don't think there will be a difference there.
Maybe. Okay. That is the galactosyl transfer is a key enzyme for making the glycolipids. So the
Yes. Yes.
So perhaps maybe there are some mutations in these particular enzyme or amino acid content may be different.
Yes, maybe there in that part. So that's more of the protein. But if you notice in the mycobacterial cell wall, there are actually no proteins, only glycans and lipids.
Okay. Ma'am, could you share some of information of Philippines dot program?
I'm sorry.
TB dot program of Philippines.
Oh, okay. So yes, so actually, so now that's our we still have a lot of cases, high cases of TB, and we also have the high incidence of MDR TB and XDR TB. Right now, yes, the another university, it's also University of the Philippines, but it's called University of the Philippines Manila. They're the ones that they are medical doctors, and they're the ones working more on the dot. So for me, I'm my focus is really in coming up with a potential inhibitor of the enzyme, and to come up with a new drug to fight tuberculosis.
That's all. Thank you, madam. Thank you so much.
Hello, Professor Gladys, madam.
Yes.
You have given a very nice presentation. And one audience have one question. Simple question is there on the live streaming chat box. I have seen the question. Dr. Govinda Bera is asking, can we use different monosaccharides unit for the synthesizing polysaccharides?
Can we use we actually tried. Yes, you can actually use other monosaccharides as long as although the based on our computational studies, the compound that we still prefer is the galactose in its furanose form. But there are papers that have reported that some of the synthesized inhibitors were actually galactose in its furanose form. So yes, definitely, I think you can try other monosaccharide units, but you have to, before synthesizing them, maybe it's better to do computational studies so that you can check if whether it will bind or not to the enzyme.
Okay. Hello, over to Bodke, madam. Hello, Bodke, madam. Hello.
My apologies if I extended. I think I spent more than 40 minutes.
It's okay.
Oh, there is a question in the chat box.
No more question.
No more.
I have not seen. Hello, Bodke, madam. Hello.
Oh, can I just sorry, I saw this question in the chat room, a chat box where it says, does the L form and D form of the
कशाला शेअर करायचं तुम्हाला काहीच नाही करायचं आहे. अच्छा ओके. पण बोलता तर येते ना त्यांना? नाही त्यांना मला ते म्यूट केलं असेल तर बाबा पाहा. Okay, madam, stop the sharing, okay, so that we can. Yeah.
Okay. Okay.
Ma'am,
Am I audible, sir?
Yeah, madam, yes.
Okay.
Hello. Bodke, madam.
Am I audible, sir?
Yes, madam. Yes, start.
Thank you so much, ma'am, for sharing your views. I am very much thankful to you from the Department of Chemistry, Guru Nanak College of Science, for sharing your deep knowledge, which is very beneficial and valuable to the society. Ma'am, focus on the glucopyrrolases linkage, which are present in the Mycobacterium galactan and its hypothesis, oligosaccharide as a substrate of and mycobacteria. Ma'am also synthesize the various disaccharide, trisaccharide, tetrasaccharide, which are used as an Gel Fd2 activity and inhibitions. So thank you so much, ma'am, for accepting our invitation and you are giving your presence to here. Thanks, ma'am. Thanks a lot. So I think technical session 1 is over here. So may I switch off toward the technical session 2. So we start our technical session 2, and so I am inviting Dr. Dinesh Deshmukh sir for the introduction of and speaker 2. So please, sir.
A very good afternoon to one and all. I am Dr. Dinesh Deshmukh, Assistant Professor of Chemistry, Guru Nanak College of Science, Ballarpur. Welcome you all in second technical session of virtual international webinar on multidisciplinary research. It's my honor to introduce our distinguished speaker, Dr. Tofida Mohammad Kabir, for this technical session. Sir is presently working as a lecturer at Federal College of Education, Zaria, Kaduna State, Nigeria. He was associated with Nigerian Army School of Education, Sobi Barak Horin, Kwara State, Nigeria. Sir has obtained his master's and PhD degree in mathematics from Ahmadu Bello University, Zaria, Nigeria. He has conducted many extramural lessons for students of Cherubim Seraphim College and GSS Rimim Doko Kaura, Zaria, Nigeria. Sir is a member of various professional bodies like Mathematical Association of Nigeria and Nigerian Mathematical Society. His area of research is pre-convection, homotopy perturbation method, thermal conductivity, viscous dissipation, and variable viscosity. Sir has published 14 research papers in national and international journal of repute and presented many research papers in national and international conferences. He has attended many workshops and seminars. Dr. Kabir delivered many invited talks in various webinars and conferences. So now I request Dr. Tofida Mohammad Kabir sir to enlighten us with talk on effect of viscous dissipation on steady natural convection fluid flow in vertical plates. So please, sir.
So we start our second session. So may I invite Professor Tofida Mohammad Kabir for the technical session 2.
Hello. Hello. Hello, Professor Kabir. Hello. Hello, Professor Kap.
I think Gurnule sir is joining him, so we have to wait for some time. Dr. Tofida Mohammad, Federal College of Education, Nigeria, who is going to deliver the lecture on the topic that is effect of viscous dissipation on steady natural convection, outlet flow in vertical plane. Participant, we have to wait for some time for the sir.
Hello, madam. Hello. Hello, Professor Tofida. Hello, Professor Tofida. Hello. Madam, sir has joined, but voice is not audible. Hello, Professor Tofida. Hello, Professor Tofida. Madam.
Yes, sir.
Sir is not audible now.
Yes, sir.
Sir is not audible.
Yes, sir.
Tofida, sir. Okay.
Yes, sir.
So just a minute, okay?
Okay, sir. We are waiting for.
Yeah. Hello, madam, ask for the Tofida, sir. Hello, Professor Tofida. Hello, Professor Tofida, sir. Hello, madam.
Yes, sir.
Tofida, sir, I think what is some problem may be there. So I can start my presentation, okay?
Okay, sir.
Okay, so then he joined, then we can take the third one, okay?
Okay, sir.
Yeah.
So, due to some technical issue and some personal problem of an Tofida sir, let us switch over the third technical session, which is to be delivered by the Professor Gurnule sir. Prior to that, may I invite Dr. Manisha Ju today, Assistant Professor Guru Nanak College of Science, for the brief introduction of an Professor Gurnule sir. Please, ma'am.
Thank you, ma'am. Thank you, ma'am. Good afternoon, one and all. I, Dr. Manisha Today, Assistant Professor in Chemistry, Guru Nanak College of Science, Ballarpur. I welcome you all in the third technical session of today's international webinar on the multidisciplinary research organized by Department of Chemistry, Guru Nanak College of Science, Ballarpur. It's my immense pleasure for me to introduce our distinguished speaker of third technical session, Professor W.B. Gurnule sir, who is going to talk on the topic nanotechnology and its impact on society. This is the subject in which we should all be deeply interested. I welcome you, sir, on behalf of Department of Chemistry, Guru Nanak College of Science, Ballarpur. Dr. Gurnule sir, presently working as Professor in Department of Chemistry, Kamla Nehru Mahavidyalaya, Nagpur. He has completed his master's degree in the chemistry in the year of 1989. He obtained his PhD degree in the year of 2000 on polymer chemistry from RTO Nagpur University, Nagpur. His area of research work is synthesis, characterization of nanomaterials, as well as development of SBR, nanocomposites materials, semiconducting materials, ion exchange resins, biodegradable
and conducting polymers, TLC and epoxy resins. Sir has published more than 200 research papers in national and international reviewed journals, and also presented more than 85 research papers in national and international conferences. Sir is honored with various awards, like he has received CRSI Best Teacher Award in the year of 2011 for outstanding contributions in the polymer materials. This award was presented by Chemical Research Society of India, Bangalore at CRSI Symposium at Bhubaneswar in the year of 2011. He has also received Distinguished Scientist Award 2016 for the contribution in research work at Chennai by Center for Advanced Research Chennai. Sir has also been received above International Outstanding Research Award 2016 by ACS Publications Salem. He also received Best Researcher Award by Vishwa Shanti Society at MDI Singapore in the year of 2017. He has received Best Research Paper Presentation Award for 6 research papers in conferences. Presently, Sir is reviewer of 33 national and international journals of repute having an impact factor more than 2. He has published more than 360 science articles in daily newspaper for people of society. Sorry for inconvenience. He has published more than 360 science articles in daily newspaper for people of society aimed to popularize the science. He has successfully guided 22 students for PhD degree and 6 are registered for PhD under his able guidance. 21 MPhil students completed their dissertation work under his guidance. Sir has successfully completed 2 research projects funded by UGC. He is the author of 32 books, chapter 3 of which are international level, published by Apple Academy Press, INC, Canada. Sir is the Secretary of Society for Promotion of Material Science, Nagpur. He is the Executive Member of Association of Chemistry Teacher, Mumbai. He is a light member of more than 12 various academic societies. His name is included in 30th, 32nd and 33rd editions of Marcus, which is the Science and Engineering USA. Sir is recently honored by Best Researcher Award given by RTM Nagpur University, Nagpur on 5th September 2018. Also awarded with Silver Medal by ISCAS Jammu on 20 December 2019. He has delivered more than 40 talks in national and international conferences. Sir has organized 6 national and international conferences for researchers of India and abroad. Sir has visited many more foreign countries like Malaysia, Singapore, Indonesia, Thailand, Nepal, Hong Kong, and Bangladesh. With this brief introduction, I would like to request respected Professor Gurnule sir to deliver his talk on the topic nanotechnology and its impact on society. Thank you.
Hello. Okay, madam.
Yes, sir. Dr. Tofida Mohammad Kabir, Federal of Education. Sir is available, sir.
Hello.
Shall we proceed for the technical session? Sir, shall I proceed for the technical session 2 or 3?
Hello, Professor Tofida, sir. Hello. Hello, Professor Tofida, sir. Can you share this? Hello.
Mic unmute karo.
Sir, unmute yourself. Sir, unmute the mic. Hello. Tofida ji.
Mic unmute करायला सांगा.
I think sir mic is unmute.
Hello, Tofida sir, unmute the mic. Hello.
Hello?
Yeah, sir. Hello.
Can you hear me, sir?
Yeah. Now you are audible. Yeah. Now you are audible. Okay.
Okay, sir.
Yes, fine. Okay.
Okay.
Sir, can you share your presentation?
Presentation?
Can I share?
Yes, I sent it through your mail, sir.
Can I share?
I sent through your mail, sir.
Okay.
Hello?
Yeah, okay.
Okay. Hello?
Yes, sir.
I sent it through your mail, sir. Hello?
Yes,
Hello?
I'm sharing. Yeah.
Okay, can I continue, sir? Hello, sir.
Yeah.
Hello? Hello?
Yeah, sharing, sir.
Okay, can I continue, sir?
1 minute. I'm sharing your screen.
Okay.
Your presentation. 1 minute.
Okay, sir.
PDF. Sorry, Okay, is it visible now?
Okay, yes, sir.
Yeah,
Sorry,
okay.
sir.
Yeah.
Okay. So can I continue, sir?
Yeah.
Okay. So good afternoon, all. My name is Tofida Mohammad Kabir, Federal College of Education, Zaria, Nigeria. I'm here to present a paper titled Effect of Viscous Diffraction on Steady Natural Convection Quid Flow in a Vertical Channel. From the title, we have the viscous diffraction, natural convection, and quid flow. What is viscous diffraction? Viscous diffraction is the transformation of kinetic energy to internal energy due to viscosity. While natural convection, when fluid flow is induced by differences fluid densities which are due to temperature change, the concentration change, or both, the flow is called free or natural convection. And the quid flow. This is a flow between parallel vertical plates where one plate is moving and the other plate is stationary. It is of two types, the plane quid flow and generalized quid flow. In the case of the quid flow, the pressure gradient is 0. So now the outline of the presentation, we have the introduction, abstract research gap statement of the problem, aims and objective, research methodology, mathematical analysis, result and discussion, then conclusion. Introduction. Fluid flows are of vital interest in almost every sphere of life. This is completely attributed to its wide application in various fields. These applications are found in the field of engineering, geophysics, environmental dynamics, architectural design, and biological sciences for better understanding of researches and application. Fluid through vertical channel is of great importance in many technological plots. Examples of these are found in the design of nuclear reactors, cooling of electronic equipment, heating of buildings through trunk walls, ventilating system, cooling and distillation stores, solar energy system, and many chemical processes. Others are found in the solid area of solid matrices exchanger, nuclear waste disposal, thermal insulation, aerodynamic heating. In fluid mechanics, convection is the transfer of heat from one place to another by the movement of fluids. Convection is usually the dominant form of heat transfer in liquid and gases. There are different types of heat transport by which convection takes place. This could either be natural convection, forced convection, and mixed convection. Natural convection occurs only by density difference in the fluid due to temperature gradient, concentration gradient, or both. The driving force for natural convection is buoyancy, a result of difference in fluid densities. Natural convection is not generated by any external source. So now the abstract. The steady natural convection flow of viscous diffusion in a vertical channel was studied. The dimensionless governing describing the different flow situations have been formulated and solved analytically using homotopy perturbation methods. The velocity and temperature profile are presented graphically for various values of physical parameters. Estimated values of skin friction, rate of heat transfer, mass plus and mean temperature are presented numerically. These parameters of interest include the viscous diffusion, the heat generation absorption parameter, Prandtl number. During the course of investigation, it was found that fluid velocity and temperature increase with an increase in viscous diffusion. Greater viscous diffusive heat causes rise in temperature, which increase the buoyancy force. It is also interesting to note that the thermal boundary layer becomes thinner, is more in viscous diffusion. The aim of this work is to study the flow behavior. So the research gap among the review literature of interest to this paper in the work of Jha and Ajibade that study the quiet flow of an incompressible fluid in a vertical channel. It is, however, observed that the work neglected the viscous diffusion and other fluid properties which are assumed to be constant, whereas in real life situation, there are a lot of applications involving viscous diffusion, and this is found in nuclear reactors and those dealing with the liquid metal, purification of crude oil. Hence, the result in Jha and Ajibade 2010 is expected to be either overdetermined or underdetermined. A statement of the problem. Due to its wide application in various disciplines, it has become very imperative to understand the behavior of natural convection in a vertical channel. As observed and pointed out in the research gap, the work of Jha and Ajibade neglected the viscous diffusion effects were assumed to be constant. However, temperature of the moving fluid affected, and it is affected by viscous diffusion. This paper is therefore remodeled to capture a real-life situation by considering the effect of viscous diffusion. The aim and objective. The aim of this work is to study the flow behavior of natural convection, an incompressible fluid flowing in a channel formed by two vertical parallel plates subjected to the boundary condition. This aim shall be achieved through the following objective, which is to examine the effect of viscous diffusion on steady natural convection plate flow of heat-generating fluids in a vertical channel. The research methodology employed in this investigation involved extension and modification of existing mathematical models and boundary conditions to capture the desired fluid properties as well as boundary conditions. Homotopy perturbation method will be used to solve the non-linear and coupled system of differential equations that govern the flow formation. Generate the line graph and numerical values of the results using computer package, interpretation of the graph so as to discover the effect of each of the governing parameters. Lastly, to validate the obtained results and draw the conclusion. Now, the effect of viscous diffusion. This is the schematic diagram of the problem. We have the heated plate and the cold plates. Under the usual assumption of the Bosnick's approximation, the governing dimensional equation of the momentum and energy for the flow formation of heat-generating fluid with viscous diffusion effect are that is equation 1 and 2, with the boundary condition equation 3. Due to the nature of quantities that are given in different dimensions, we introduce some dimensionless quantity that can transform the governing equation and their condition into dimensionless form. The dimensionless quantities used in equation 1 and 2, and the boundary condition 3 is here in equation 4. By using the dimensionless quantities, the governing equation and the boundary condition are transformed into non-dimensional form, that is equation 5 and 6, that is the momentum and energy equation, and the boundary conditions 7. In order to solve equation 5 and 6 under the boundary condition 7 using homotopy perturbation methods, we introduce the convex homotopy as, that is equation 8 and 9, for the momentum and the energy equation. The zeroth order is approximation is linear in the present problem. Therefore, the use of initial approximation is not required. When zero orders is not linear, then there is need for initial approximation. Assume the solution of equation 5 and 6 to be written as equation 10, that is velocity and temperature. Substituting equation 10 into equation 8 and 9, we have equation 11 and 12. By comparing the coefficient of P0, P1, and P2 of equation 11 and 12, we have equation 13 up to 19, that is P0 for velocity and P0 for temperature, up to equation 19, up to equation 20. And the boundary conditions are transformed also as equation 21. So be equation 13 and 14, and applying the boundary condition for velocity and temperature, we obtain equation 22 and 23 as this. Now, so be equation 15 and 16, and applying boundary condition U1 of 0, which is 0, and U1 of 1, which is 0, and T1 of 0, which is 0, and T1 of 1, which is 0, we obtain equation 24 and 25. Now, solving equation 7, 17, and 18, and applying the boundary condition U2 of 0, which is 0, and U2 of 1, which is 0, and T2 of 0, which is equal to 0, and T2 of 1, which is 0, we obtain equations 26 and equation 27. Equation 22 to 27 give the expression for velocity and temperature as equation 28. That is, now don't forget the homotopy perturbation is a series solution. That's why U equal to U0 plus U1 plus U2 plus U3, and T, that is the temperature, which is T0 plus T1 plus T2 plus T3. Now, where the constant, that is A1, B1, which is -1, A2, B2, which is 1, and A3, A4, and A5. To obtain the skin friction and rate of heat transfer of the channel boundary, the expression for velocity and temperature are differentiated with respect to T, that is du dy, which is at y equal to 0 and at y equal to 1. Now the skin friction means is the friction between the free flow and the resistance. Now equation 22, now this is equation 29, that is skin friction at the plate y equal to 0, and equation 22, equation 30, which is the skin friction at the plate y equal to 1, and the rate of heat transfer at the plate y equal to 0, and the plate y equal to 1. That is the heated plate and the cold plate. We further obtain the max flux Q and mean temperature by evaluating the integral from 0 to 1 for velocity with respect to y, and mean temperature by multiplying velocity and temperature divided by velocity from 0 to 1. That is equation 34. Results and discussion. The present work analyzes the viscous diffusion effects on steady natural convection fluid flow of heat generating fluid in a vertical channel. The velocity and temperature fields are presented graphically in figures for various values of heat generation, absorption, parameter, that is S, Grashof number, that is GR, Prandtl number, PR, and viscous dissipation, which is AC. For the purpose of this discussion, the parameter of interest are arbitrarily selected between 1 is less than or equal to GR, but GR is less than or equal to 9, and S is less than or equal to 2, between negative to positive 2, while the working fluid is considered to be air, so that PR, which is equal to 0.71. Now, figure 2 and 3 display the velocity and temperature profile for different values of heat generation absorption parameter. It should be noted that the positive values of X signifies heat generation, while the negative value signify the positive values of X signify heat absorption, while the negative values signify heat generation. It seems from the figures that as the heat generation increases, fluid temperature and velocity increases, while it decreases with increase in heat absorption. Increasing the heat generation amplifies the applied boundary heat, which causes the fluid temperature to increase, and as such convection currents, and as such strengthening the convection current within the channel, which is turned in increasing the velocity. In addition, fluid temperature drop as a result of increasing the heat absorption parameter, and the thermal boundary layer becomes thinner, thereby reducing the velocity distribution of the working fluids. Now, figure 4 and 5. The effect of viscous dissipation on the fluid velocity and temperature is depicted. It is interesting to note that fluid velocity and temperature increase with an increase in viscous dissipation. Greater viscous dissipation heat causes rise in the temperature, and as consequence increase the buoyancy force, so that fluid velocity increase as viscous dissipation increases. It is also noticed that the thermal boundary layer thickness is more in the presence of viscous dissipation. Now, table 1 is the estimated numerical values of the skin friction at the plates y equal to 0 and y equal to 1 for various values of PR, that is Prandtl number, and EC, which is 0.2, 0.4, and GR, that is the Grashof number, which is 5.0 and 8.0. The skin friction in figure 1, the skin friction of the boundary surfaces of the boundary plate is simulated and presented in table 1. From the table, it is seen that the skin friction is higher when the working fluid is air as compared to that of mercury. PR, which is 0.044, is mercury, and PR, which is 0.71, is the Prandtl number of air. That's why the skin friction is higher when the working fluid is air as compared to that of mercury. If you see on the table, we have 0.83132 from PR, which is 0.71 at the heated plates, and 0.80079. So therefore, 0.83132 is more than 0.80079. The table further shows that growing viscous dissipation, heat source, as well as the buoyancy parameter have tendency to increase the skin friction on both boundary surfaces. However, heat sink contribute a decrease in the skin friction, and this due to velocity decrease caused by growing heat sink, which consequently lead to decrease in the skin friction in both plates. Now, table 2. A clear view from table 2 shows that as Prandtl number increases, the rate of heat transfer decreases, on the heated plates, while it decreases on the cold plates. This is due to the fact that temperature increases with the increase in PR, leading to decrease in the temperature gradient on the heated plates, and the reverse trend also observed on the cold plates. Furthermore, growing viscous dissipation, heat generation, as well as buoyancy parameter lead to a significant changes in the rate of heat transfer. This can be attributed to decrease on the heated plates, while the reverse trend is observed on the cold plates. While, however, heat absorption leads to increase in the heat transfer on the heated plate, and this due to the temperature decrease caused by growing heat absorption, which is consequently lead to increase in the rate of heat transfer on the heated plates. This is physically expected since fluid temperature increases with increase in heat generation, and this decrease the temperature gradient on the heated plates, while heat transfer to the cold plate increases. Now, table 3 present the max plus for various values of PR, EC, and GR. It is clearly seen that the max plus increases with increase in heat generation and decrease with increase in heat absorption. The table further shows that the max plus increases with increasing values of viscous dissipation and Grashof number. In addition, growing Prandtl number is also observed to increase the max plus. The max plus is shown to be lower for mercury as compared to that to air, because air possesses higher temperature and velocity than air. Increasing Prandtl number constitutes growth in the temperature because of lower diffusivity, which strengthening the convection current and velocity increase, so that the mass plus increase with growing PR. Now, table 4 present the numerical values of mean temperature with various values of PR. It is observed from the figures with the increase in heat generation, mean temperature increase, and the opposite trend observed in heat absorption. Furthermore, growing Prandtl number leads to increased mean temperature. The table further shows that the mean temperature decreases with the increase in buoyancy parameter. This shows that the influence of Grashof number, the influence of buoyancy on the mass plus, overshadow its influence of the bulk temperature. Now, the final table, that is table 5. This is the numerical comparison between the work of Jha and Ajibade and the present problem. Jha and Ajibade here, so the validation. By setting the viscous dissipation parameter to 0 in the present problem, we recover the result of Jha and Ajibade. The comparison is presented in table 5. Now, the conclusion. The present problem has considered the viscous dissipation effect on steady natural convection wet flow of heat generating fluids in a vertical channel formed by two vertical parallel plates. The work concluded that velocity and temperature profile increases. Velocity and temperature profile increase with the increase in viscous dissipation. As the heat generation increases, fluid velocity and temperature increase, while it decreases with the increase in heat absorption. Fluid velocity and temperature increase with growing Grashof number. Velocity and temperature profile increase with the increase in Prandtl number, and also found that the skin friction is higher when the working fluid is air as compared to that of mercury. Furthermore, mass plus increases with increasing values of viscous dissipation and Grashof number, while an increase in the buoyancy term constitutes a decrease in mean temperature within the channel. In addition, it is interesting to note that the comparison made between this work and that of work of Jha and Ajibade showed that the present work agrees significantly with the Jha and Ajibade 2010. Finally, it is concluded that viscous dissipation is very significant in natural convection problem and should not be ignored in future investigation. So this is the reference. Thank you.
Thank you so much, sir. Thank you so much, sir.
Thank you, ma'am.
Participant, thank you so much. Hope you will be listened to you further in the future, sir. So, sir covered the various effect of viscous dissipation on the steady natural convection flow in vertical plates and its significance in natural convection. So, thanks so much, sir. Participant, if you have any doubts, please share, put it in the chat box. Participants, if you have any doubt or queries regarding this session, please put it in the chat box. I think no question from the participants. Sir, any doubt or questions? I think no question from the participants, sir. So, thank you so much, sir, once again. I'm very much thankful from the Department of Chemistry for delivering your lecture on such a nice talk. Thank you, sir. May we switch on the third session, sir? Technical session third will be started. So, may I request Gurnule sir to please share the screen for the technical session 3.
Thank you, madam. Thank you for the yes, madam. Thank you for inviting me for this international webinar. So I am thankful to the organizer for the invitations. Today I'm going to discuss the part nanotechnology and its impact on the society. Before going to start, before going to start, I again now thanks the principal of the college, Dr. Bahirwa sir, Dr. Khobagade madam, and all the faculty members for giving me the chance to give the talk in this international webinar. So title is the nanotechnology and its impact on the society. So here we are discussing different aspects about the nanomaterials and nanotechnology. As we know that today we are in a dynamic world where time domain and the geography are rapidly shrinking, but the space and opportunity are expanding with the competition and innovation as the key driver. As we know that knowledge is a driven economy. We know that knowledge driven economy, which is linked with the science and technology. As we know, science and technology is these are the two terms, and these two terms are different, but at the same time they are complementary with each other. Do you know what is science? Science is the pursuit of knowledge and understanding for its own sake, and technology is the creation of artifacts and the system to meet the people needs. And therefore, we can say that the development of many countries based on that make our life depends upon the it make that our our life make a comfortable and which is related to the materials. So development of many materials are there, and these materials are giving us, that is our making the life comfortable, and this is related to the materials. So as we know that there is one discipline of material science, that is the discipline of material science is the interdisciplinary and also it gives the relation between the structure and the properties of the material. Now there is also material engineering, and in where they can give the design to this material as per the requirement of the society. As we know that material science and material scientists play a very important role in the development of the country. Properties of materials are the size dependent, and also it is shown that it is now known that material scientists claim that 21st century is the century of materials, and especially the nanomaterials. So now, before going to discuss details about that, as there is a revolution in the industrial, and there is a change as the time goes. After the Middle Age, the steam engine came. After that, railways came. Then automobiles. So computer came. So step by step, there was changes as the time progressed. And after this, the next big step is the nanotechnology. And so we should know what is the nanomaterials or nanotechnology. So there is now, we can say that there is if you see the year-wise from 1970 up to 2020, there is a low, there is opportunity and also high research was there. So in the beginning, there was a basic research. After that, applied research came, and then early adapter. And now after 2020, we can say that there is a mass market about the nanomaterials or nanotechnology. So in this case now, one scientist that he that is the scientist says that this nanotechnology is not new. So it is ancient, and therefore the contribution of elemental carbon to the development of nanoscience and technology at the Indian Institute of Science, Hyderabad, the Nobel Prize winner, that is the Robert Kerr, says that carbon nanotechnology was not much older as compared to carbon nanoscience. He also says he got a Nobel Prize in chemistry in 1996 for the discovery of fullerene, along with the Richard Smalley and Howard Cotto. He also says that, and he has carried out some analysis, and also says that the sword, that is the king in the old age used for fighting with the enemy, that is the Tipu Sultan, or what is we can say Maharana Pratap or Shivaji Maharaj. The sword also contains some large number of carbon nanotubes, but people does not know that contains the carbon nanotubes or carbon nanomaterials. Similarly, the glass material in the Roman times also had nanomaterials, and they also said that glass was covered with the nanoparticles of the gold or silver. And he said that people were using this such material of nano size, but could not, they does not know, they do not have understanding what they were doing for the long time. Now this is the is not nanomaterial, is not ancient material. Ancient material is not a new. So now here, this is the daylight inside and light. Here, this is the Lacquer's cup, which is kept in a British museum. This is the Roman pot, and here here red color is nothing but the is from the nano size gold. And this paper was have been published in Nature in 2000, and says that this nanotechnology is a ascent material they are using. So this is the in daylight, see the color, and in a light they show the light is emitted, that is the red color. So the red color is nothing but the nano size gold. Now what is now before going to so first of all we have to know the what is nano materials. So in this case we know that what is mean by nano. As we know, nano size means the word nano is taken from the Greek word nanos, meaning dwarf. It is used to describe the 1/1,000,000th of the something. And as we know, 10 to the power -9 is the nano. And we can convert this 1/1000 meter equal to 10 to the power -3 meter and millimeter. So this is the conversion how 1 nanometer is there. So micron to meter and meter to nanometer. So 1 nanometer means 10 to the power minus 9 meter. So in this way, this is the scale also shown here, how to convert centimeter, millimeter up to nanometer. So this is the scale to convert in nanometer. Now how to represent? So this can be represented in this way, 10 to the power minus 9.0009 here, 00 and then 1 meter. So in short, we can say that 1 billionth of meter is 1 nanometer. 1 nanometer is approximately 6 carbon atom wide. A hydrogen atom is 0.1 nanometer. And human height is a 1.7 billion nanometer. So this is the representation and the in nano size. So what is now nanoscience or nanotechnology? So in this case, particles of the material less than 100 nanometer in diameter are said to be nanomaterials. And study of such type of science or material is known as the nanoscience. So particle size less than 100 nanometer is the diameter is said to be nanomaterial, and study of such material is the nanoscience. And technology is there, and it is the branch that give the design and characterization, production of this material, and for application of particular purpose by controlling the shape and size of material. They are designing as per the requirements. So there is a nanotechnology. So what is nanomaterials? What is nanoscience and nanotechnology? So we can say that here, this is science at the nanoscale. As we know, this material science is an interdisciplinary science which is related to the chemistry, physics, biology, material engineering, earth science, computer science, and so many fields are there where there is a interconnected, and therefore we can say that it is interdisciplinary science. The power and now there are a number of applications of this technology because based on the research and on the technology design, there are a number of applications in the healthcare, for energy, and also for the environment and technology. Now, nanoscience and nanotechnology has a great potential, revolutionary potential, and will have significant economic benefit. But at the same time, there are some ethical, legal, social, and environmental issues to be addressed. And therefore, an integrated and responsible approach is shown here. That is, nanotechnology requires action at several fronts. As we know here in this slide, research and development is there in industry and some of the industries, and also in research institute, where we require infrastructure, human resources, industrial innovations are there, societal issues are there, and therefore, because when we deal with the nanomaterials, then we should have to take the care of about the health, safety, and environmental and consumer protection. And therefore, here at international cooperation should also be required, and therefore international level we have to take the some that is the care about health, safety about this nanomaterial in case of the R&D section. Now there are two approaches. One is a top-down method and one is a bottom-up method. These are the two scientists. They got a Nobel Prize. First scientist Richard Feynman. He is a physicist and got a Nobel Prize in 1965 for the fundamental work in quantum electrodynamics. And this Richard Feynman says that there are plenty of room at the bottom. What is the first approach? Is the top-down approach. Top down. So we have to start from bulk and we get the nanomaterials. So this is the top-down approach. And so Richard Feynman says that there are plenty of room at the bottom. That is in case of the nanomaterials. It is a small, very small particle, and also according to that, there are plenty of room at the bottom. Means that suppose you know that one room is there, we can divide by the room in two parts. We can also divide it into 4 parts, also as per the requirement. So such type of plenty of rooms are there in the nanomaterials. We can make the compartment as we required, and therefore this is very important material for the scientists and also for the society. And that is why there are plenty of at the bottom. Second approach is also there, bottom up. And Jean-Marie Lehn also received a Nobel Prize for the development and use of molecule with structure-specific interaction of high selectivity. And Jean-Marie has also suggested that there are plenty of room at the top. So in case of bottom up, start from atom to atom, start from molecule to molecule, and we get the nanomaterial. So it is known as the bottom up. So he says that there are plenty of rooms at the top. So these are the two great scientists. They have received the Nobel Prize and also given the top down and bottom up approach for the nanomaterials. Now, thing is that why do we want make the thing small? So we can say that we make the thing small because it should be cheaper, smaller, faster, better, and easy to handle, and also easy to carry and use in the different purposes. And therefore, we want the thing small. Also because to introduce the new physical phenomena for science and technology. So that is why we want the things small. And now here we can say that what is the nanomaterials. When we discuss about this material, with the change of structure, there are changes in a different chemical and physical properties, and depending upon the size of material. In case of small material, in which strong chemical bonding present, where delocalization of electrons are extensive and extent of delocalization vary with the size. So they are property dependent, and also size play an important role. So in this case, now changing the size or properties in a nano region. So here three parts we have here it is given. One is a quantum mechanical behavior, in which we say that it is a new technical physics, and because of that there are changes in color, transparency, hardness, and electrical conductivity, which is shown. And second, increase the surface area, and it is a chemical process, and show the changes in melting and boiling point, chemical reactivity, catalytic activity, and also in third, molecular recognition in biological science. So bio applications are there. So by the combination with the cell organization, repairability, adaptability, recognition. So all these are the three important part. As we change the properties in the nano, they show the changes in many properties, and these three phenomena are given here. Now the important part of this nano size. So nano size causes the changes in, these are the 7 properties: color, conductivity, up to, and all these are the 7 properties where nano size show the change in properties. So this is an important, as we know that nano size gold, as we have seen, we know that gold, when it is in a bulk, how the size changes, the properties also changes. So in this part, it is shown. This is the what is the gold, bulk gold, having melting point 1064. Color is golden. We have seen the golden, but now as the size changes, they show the change in color. So here, first here, 1 nanometer gold particles have this color. What is this? Brownish yellow color. And if the size is 20 nanometer, melting point, lambda max also changes, and color is red. And this is the purple color pink, or 100 nanometer. So as the size changes, 1 nanometer, 20 nanometer, 100 nanometer, they show gold, they show the change in properties. And also here, when it is in bulk, they show the that is the golden color. So now the property changes, and this property depends upon the what is the size. So as the size change, color also changes. So this is the important that gold show the different colors. So we know it is golden, but now if the size change, the color of the gold is also changes. These are some Indian phosphorus nanomaterials, cadmium, selenium in solution. So you see now here color, how they are changing, the size changes the color. So if the nanometer is 4, 4.2, 4.8, 5.5, so cadmium, selenium also show the changes in color. So this is an important, as the size changes, color also show the change. And now there is a this slide shows that nanosizing changes the chemistry. We cannot change the chemistry, but now nanosize can change the chemistry. And here, this is the metal oxide of gold surface. And here, the reaction is shown here that when the material gold is larger in size, when the reaction is taking place, then what is oxidation? So what is now here? There is an emission of carbon monoxide only. They give only carbon monoxide. But when the size is small, they give the carbon monoxide along with the carbon dioxide. So material is same, but they show some different product. So it depends upon, and this paper have been published in a European chemical journals by the CNR Rao. So synthesis of nanomaterials are the important part. And so in case of synthesis, two methods have been given here that we have seen that one is a top-down and bottom-up methods. So first method is a top-down and second is bottom-up. So top-down, in case of top-down, we have to start from the bulk, cut it, the bulk material cut or slash it in a properly so that we get the nanoparticles. Or in a bottom-to-up approach, here we have to take the atom and atom, molecules and molecules, or cluster and clusters, and then we get the nanoparticle sheets here. So these two methods are there, two approaches are there, that is bottom up and the top down method. So these are the two important methods for the synthesis of nanomaterials. In addition to that, number of methods now available, that is chemical methods, sonochemical routes, mechanical, lithography, etching. So microwave assisted method is also there. So different methods are there, and some modern methods are also there. Sol gel method we are using, chemical precipitation, hydrothermal method is also good. And so these are the different methods, modern methods are available for the synthesis of nanomaterial. Now one method is given here, that is the sol gel method, and this method consists of 7 steps. One is first is a mixing, then casting, then gel gelation, aging. So these are the step by step 7 stages have been given, and lastly we get the in a sol gel process dense gel. So this process is a wet chemical technique used for the fabrication of both glassy and ceramic materials. In this process, use of either chemical solution, that is sol solution, or colloidal particles, sol for nanoscale particle, to produce the integrated network, that is gel. After drying process, the liquid phase is removed from the gel. So this was strongly advocated by the Heyman is the scientist, and adopted by this and also published the paper. Sol is a liquid in colloidal solution, and gel can be described as a solid or semi-solid. Sol can be converted into gel by cooling, and gel can be converted into sol by heating. So this is simple method one can carry it out. One can one can do this method for the synthesis of simple metal oxide of nano size. And next is the solvothermal. By using the solvent also, it is possible to synthesize the nanomaterial. And solvothermal method, this method is a typical wet chemical synthetic strategy using water or organic solvent as a reaction medium in a sealed vessel, that is autoclave, in which the temperature range is 100 to 240 degrees Celsius. This method is better than other method, and the precursor solution is usually non-aqueous. So this method has been used in laboratory to make the nanostructure tin oxide, graphene, or carbon sphere, or the other materials. So solvothermal method is also important. And another method is there, hydrothermal method. In case of hydrothermal method, can be used to synthesize the material, and this method include various techniques of crystallizing the substance from the high temperature aqueous solution to the high vapor pressure. And this is the method of synthesis of single crystal that depends on the solubility of minerals in hot water under high pressure. The crystal growth is performed in the autoclave, in which the nutrient is supported or supplied along with the water. Temperature gradient is the maintained, and at a hotter end, nutrient solute dissolves, while at the cooler end, it is deposited on a seed crystal, giving the distorted crystal. So temperature can be used up to 300 degrees Celsius. So all these 3 methods we can use. Another method is the microwave assisted method. By using this also, we can carry out the synthesis of material. So this is some, in case of solvothermal, if you change the solvent, we get the different morphology. So this is the morphology changes with the change of solvent. In case of solvothermal, if you take the ethyl alcohol, if you take the mixed solvent, then if you take the pure water, so see the morphology show the changes. And after that, Here, after synthesis of this material, now it's important to know the methods of characterization of the nanoparticles. So same X-ray, AFM, BT are the techniques we can use for the characterization of the materials. So this is the same scanning electron microscopy can be used to know the surface morphology of the synthesized nanomaterials, and it is an electron microscope that image the sample surface by scanning it with the high energy beam of electrons, and therefore it gives only the surface, what is the nature of material on the surface that can be obtained from this same. And more detail is obtained by using the transmission electron microscopy. And here the beam of electrons, a beam of radiation, so completely passing through the whole body of the material. So what is the inside bulk in case of this material? What is the nature on the surface, as well as what is the nature of the material inside the material, that is inside. So we know the nature of the material, complete part of that material. So this is the microscope technique, whereby beam of electron transmitted throughout the ultra-thin specimen of the material and passes through the sample, and it gives the image. So this is more accurate, that we know the whether the particle or material is in a nano size or not. So another AFM, atomic force magnetometer, is an important tool or technique. We know with the help of this, the arrangement is like this. Photoelectrons are here. This is the pointer, is their tip of this here, and this is the laser beam is there. And now this is the surface of the metal oxide, and they show the nature of when this tip we place on the material, where we place it give the information about that part. We can so in this way we can know the size or nature this part any part of that material. So perfect information is obtained by using this AFM. So another method, that is the by using XRD and by using the Cherer formula also, it is possible to know the particle size and also nature of the materials. So by using this equation, we can calculate number of parameters, and so these are the methods to characterize the synthesized materials. And now after this here we can say that carbon nanotubes, whenever we are discussing the nanomaterial, then we should also know the carbon nanotubes. So it is very important, because number of allotropes of carbons are there, fullerene and other, and therefore we should have to know the details about the carbon nanotubes. So here these are the allotropes of the carbon. One is diamond, graphite, carbon nanotubes, C single walled, multi-walled, and also the graphene, and also here fullerene. So all these are the which contain allotropes of carbon and contains carbon. So now here is shown nanotube, carbon nanotube is there, and there are different types of carbon nanotubes, and one is a single walled and another is a multi-walled carbon nanotubes. So we can say that single walled and multi-walled carbon nanotubes. Single walled carbon nanotubes are defined as the one-dimensional or cylindrically shaped allotropes of carbon that have high surface area, and single walled carbon nanotubes are a special class of carbon known as the one-dimensional material. And in this case, they consist of sheets of graphene of 1 atom thick. So it is single walled only. And in case of multi-walled, which is very useful, and multi-walled carbon nanotubes are elongated cylindrical nano objects made up of sp2 carbon. Their diameter is a 3 to 30 nanometer, and they can grow several centimeters long. It is a allotropes of carbon and that have high aspect ratio, means length to diameter ratio. It is also called double or triple walled carbon nanotube. So this is the single and this is multi-walled. And in next slide, it is shown that these carbon nanotubes are having 3 different shapes. One is an armchair, second is a zigzag, and also chiral. So it is shown here. This is the carbon nanotube in picture, and here they have 3 different types of shapes. Now one method is also, we should also have to know the methods or synthesis of carbon nanotubes. So what are the methods for the synthesis of carbon nanotubes? Generally 3 methods are there: electric arc discharge, chemical vapor deposition, CVD method, and laser ablation. And here, one method I'm giving that carbon nanotubes for the synthesis of nanomaterials, carbon nanotubes. And here, the arrangement in this way is like that. Here is shown. Here is the reservoir. And here we have to pass the carrier gas argon. And ferrocene we have to take in a reservoir and dissolve in a benzene. And condition solution flow rate we have to keep the 3 ml per minute. Carrier gas flow is a 50 ml per minute. And atmosphere we can take inert air, that is the argon atmosphere. And so this is the arrangement. And from the reservoir we have to pass the solutions, so that here is the arc is there and the struck, and we get the material that is found in a carbon, that is the CNT. So this is the CVD method can be performed for the synthesis of CNT. So it is very good method for the synthesis of carbon nanotubes. And after this, now some properties are there of the carbon nanotubes. That is here CNT have some mechanical property. It is stiff as diamond. Young modules is greater than 1 terapascal. Tensile strength gigapascal equal to 200. Electrical properties, metallic, semiconducting, and thermal conductivity. So they have all these are the properties related to the carbon nanotubes. Now there is also a functionalized carbon nanotubes, and in this case, as we know, in the research we can attach number of groups to the carbon nanotubes. This is the one carbon nanotubes, and when we introduce the HNO3 and when we heat, what is then these groups get attached here in this way. OH carboxyl group can we attach, or if you add the RNH2, so yeah, RNH group can attach. So we can attach the any group, that is OH group or hydroxyl group or amine group, amide group, to the carbon of this CNT, so that we can change by introduction of the new group in the carbon CNT. They show the change in the properties of the materials. So now there are some applications of these carbon nanotubes, and these applications have been shown here. Carbon nanotubes are being used to develop the flat screen television. Nowadays, market having a flat screen, and also higher resolution is also there. That is now 4K, and now recently also came 8K like that. And your next TV screen should be could be a thin and ultra light foldable. So just we can also carry. It is so thin that we can also carry the. If it is foldable, then we can also carry that TV from one place to other easily. And thin means just like a paper. Paper is thin. In the similar way, the TV can also be a thin, and therefore this material CNT can also be used in a badminton racket, tennis racket, incorporated. And because of that, the racket weight is a very light and also very strong, and therefore it is used for that. Use of carbon nanotube in a solar cell is also now scientists are using for their greater efficiency. Branching and also switching of signal at the square. So electronic junctions, these materials now is also been using for the signal in the city. This is the slide which shows the effect of carbon nanotube, that is CNT, during the hydration of the cement. Now here, this graph shows this is the different natures of the material. This is the absence of CNT. What is the nature? See here, somewhat different. And here, if we add in the cement, only the presence of single wall carbon nanotubes, and here presence of multi-wall carbon nanotubes. So there is an effect of CNT on the hydration of here opaque materials. And here this first is the plain cement paste. When we add a single wall, then the efficiency slightly increases. And if we add the multi-wall, then see the 30% efficiency is increases. So we say that there is an effect of the CNT in the cement. So they show the increase in the hydration process. Now, as we know, after this is shown here that carbon is a wonderful material, and here nano gold is the wonderful materials. Carbon is wonderful, and here nano gold is the tiny particle of gold, and are so small which is measured in the nanometer. A nanometer is a billionth of meter smaller than wavelength of light. So gold itself does not always glitter. Now it is widely used for the electronic conductors, therapeutic agents, photovoltaics, drug delivery in biological and medical applications, and also as a catalysis. So this is a very important, nano-sized gold is very important material. Now in case of the, in this case, as we show in case of gold, catalytic properties of nano gold, it is an excellent catalyst, and it is because the smaller the size, surface area of nanoparticle become very high, and hence high catalytic power is there, and therefore it is a good excellent catalyst for many reactions. And here as the size changes also, the shape is also changes. So here two shapes have been shown. When it is a 3 to 5 nanometer in diameter, then it has a icosaedral. When it is a bulk, then it is a face center cubic. So changes of size changes the shape of that materials. So here structure also show the FCC structure in case of nanoscale gold particle, but as the size changes, the structure is also show the change. Now nano gold used as a catalyst, and we have seen this one. These are the different field where we can use nano gold as a catalyst. So very high selectivity in partial oxidation reaction from propylene to propylene oxide at 5 degrees. Gold is used as a catalyst. Selective oxidation process also where we also use the nanoparticle of gold catalyst. Gold in recent year has drawn more attention because researchers are having now using this potential catalyst in a chemical processing, also in pollution control, and also for the fuel applications. So here a reduction of nitric oxide too with the hydrogen can also be carried out in presence of the nano gold. So also in a different field we can use this as a catalyst. And nano gold in anti-therapeutic, that is for the drug delivery, we can also use. And nanotechnology provided a very good tool toward the solving the problems in biology. And important tool here is that nano medicine can be injected in the body, and it after it reaches toward the infected part, and it repair that part and then get digested because it is DNA based. So it is an important application of this nano gold. And here how this nano gold is injected. So this is the nanoparticle shown in color, and this is the nano gold single, and it is a drug loaded particles. It reaches toward the infected part and it get repaired. So nanoparticles can also be in multiple drug. We can also multiple types, that is the different nano drug gold can also be injected, and after the repairing the part, infected part, it get digested as it is a DNA base. And now here silver nanoparticle can also be used sometimes for this purpose. For the different applications are there in case of silver nanoparticles, and silver nanoparticles are the nanoparticles of silver between 1 nanometer to 100 nanometer in size. Some compound contain large percentage of silver oxide. Silver nanoparticles are efficient, absorbing and scattering light, and unlike many dyes and pigment, have a color that depends upon the size. So silver nanoparticles have shown excellent bacterial property against wide range of microorganisms. And another important tool is here zinc oxide is the important part, and here zinc oxide nowadays widely used for as a medicinal purposes, and also used for manufacturing rubber, also as a pigment for paint and coatings. And there is a bright future of this zinc oxide that shows promising sign in the field of nanotechnology, EV detector, nanoscale detector, and actuators. And also it is also dual, showing both semiconductor as well as piezoelectric properties. So zinc oxide is very important material in this field. Now here is shown how to synthesize or how to make this zinc oxide nanostructure material. The most common used zinc oxide nanoparticle is in the sunscreen. Also we generally use zinc oxide in the sunscreen. They are used because they reflect the UV light. And synthesis of zinc nanoparticles was achieved by using zinc powder, polyethylene glycol, and ammonium carbonate by precipitation method. One of the simple method also given here, flow chart is given here. So we have to take this material, silica tube was preheated temperature, and after that you have to keep in furnace. After that, carrier gas and oxygen, reactive gas, and 1 gram zinc powder we have to place in alumina boat and inserted. After that, zinc oxide material from the white cotton like got after the that deposited on the boat silica tube, and after that we have to characterize, and also we can use as a after characterization nanoparticle, nano rod. We get the different shape: nano tripod, nano flower, nano comb, nano tree, nano nail. Such type of morphology is obtained in case of zinc oxide. So these are the morphology of the see the shape: nano kite, looking like kite, a nano kite here, a nano comb, a nano star, nano wires, nano comb. So different shapes are there, and by using the different methods, by changing some properties, we get the different nature of the zinc oxide. So in case of the magnetic properties now, here magnetic properties also changes with the change of size of the materials, and we can say that magnetic properties of nanomaterials or nanoparticles have diverse applications. These particles used in cellular phone, credit card, also microwave devices, and also if the size changes, then magnetic properties also show the change in the properties. So magnetic properties is very important. And now there are some benefit today and tomorrow about this. That is, we also use this material in medicine and health, also for the drug delivery. Information technology is now also been, this material can also be used. Another energy production and for the storage, and also for the material science, UV, lightweight and strong materials. For the food storage and for water, and for the environment, remediation protection, and there is a instrumentation and tunneling microscopy. So there is a importance of this material today and also tomorrow. Now there are a number of uses of this material: nanomedicine, nanosurgery. These are the paint, cosmetic services. They have been used here. And nano catalyst, nano composite show these different types of applications. Number of applications are there, but now I am giving here 3 important applications of this material. So 3 important materials of this nanotechnology or nanoscience. One is for the strain-resistant cloth, and second for the better storage of computation. Third is that chemical and biological sensors as a drug delivery devices, and for the environment, clean energy and clean air. So these are the main important part that I'm going to discuss now. First part is the material strain. What is now in case of material, strain resistant cloth are there. Nanofiber create a cushion of air around the fabric, and 10 nanometer carbon whisker bond with the cotton act like a pitch food, many liquid roll. That is, if the any dye particles are there, and nano paint that refuse this strain. So this is the material that if you now say, scientists are saying also that our fabric may also come made up of the nano fabrics, and this fabric, if there is any ink or any color pigment, if any color, then easily we can remove by rubbing it. So there is no any that is the, it may be also a dirt repelling and also the color repelling, and that is why nano care fabrics with the water can be easily removed, and vegetable oil, mustard, these can be removed easily. So this is the strain resistant cloth. So it may come in a feature that we also have a, we may wear the strain resistant cloth. So it can remove dirt, it can remove the color of the paint easily. And second important application of this material is the paint that does not chip. And now our car having this protective nano paint. So nano paint can be used for the car, and it is a very shiny, and nano, after using this nano paint, give the shiny color. Also it is a self repairing. If there is any crash, it can be repaired. Also, now scientists are using that the color of the car can also get changed automatically. So water and dirt are repellent. Another that resistant to chipping and scratches can be removed, and also brighter the color, enhance the gloss. And in future, it is also now shown that change in color and self repair. So car can be changed as per your choice of the color, and so in the future it may come that one car can show the change. The materials are going on in this way. Research is also going on this part. And another application of this material is that in the environment to reduce the pollution and paint that clean the air. As we know here in the figure shows Burj Khalifa in the what is the tallest building in the here it is shown, and this building outer side is coated with the tin oxide nanoparticles. This tin oxide present in the as a nanoparticle in the paint breaks down the organic and inorganic pollutant that wash off during the rain. So it does not damage the monument or any buildings. So it decomposes also the air pollution particle like formaldehyde, and because of that, nano paint on the building can reduce the pollution. So we can say that in terms of pollution, to reduce the pollution, nano paint or nanomaterial can be very useful. Another now application of this is that environment nano solar cells. So nano solar cell can also be used, and this nano solar cell mixed with the plastic could be painted on the bus, roof, clothing, and also solar become cheap energy alternative, as we know that. And so this solar cell, nano solar cell, can be put it on the bus on the top roof, so that a bus can run on that nano solar cells. Also train in future, train may also run on the nano solar cells. Instead of using this electricity or coal, nano solar cells can be used for the running the train. Also, we have to place the nano solar cells on the top of that train or bus or house also. So this is the mechanism is shown here. They produce the heat energy absorbed from the sunlight, and there are two electrode. Nano rod is mixed with this here, and they show they absorbing all this energy and storing for that and giving the lot of energy, and which can be used, solar energy can be used for these different purposes. And here we have to mix nano materials, so nano solar cells are obtained. Another important application of this material is the in case of the of this nanotechnology is the, we can say that DVD that could hold millions of movies. As we know, this is the magnetic properties. Our current CD or DVD media have storage scale of micrometer, and if you are using nano size material, then it increases the 1000 times more storage capacity as compared to CD or DVD. Or we can say that new nano media made from the gold self-assembly into strips of silicon has a storage scale in nanometers. So 1 lakh times greater capacity is having if you use the nano with the silica. And as we know, our chips made in ATM card or debit card, also in the mobile. So what is now the capacity? Chip is very small, but having the more capacity. So in the future also, if you are using nano size in the silicon, then what is, we can also increase the capacity of the storage capacity of the materials. So here CD is shown, having CD can store one movie, DVD can store 4-5 movies, but silicon chip also can move here embedded with the nanomaterial. So having more greater storage capacity, or we can say that 1 lakh times greater storage capacity as compared to other materials. So these are the important applications of this material, and some medical field applications have been also been shown. By it can also be useful in case of biomaterials, bone, teeth, cells, immune system also for surgery. So these are the field where we can use this material. So there are 3 possible reasons for color, and here in this case, potential risk for the technology. but now here, whenever we are discussing with the nano material or nano size, there are some health issues are also there, some environmental issues are also there, and now also risk assessment is also there. So we should have to take care whenever we are dealing with this health or environmental issues. Now challenges to the research institute are some here that our ex-president Dr. APJ Abdul Kalam says that creativity leads to thinking, and thinking provides knowledge, and knowledge makes you great. So we are witness that convergence of the informatic, communication, biotechnology, and nanoscience. So here we can say that there is a part that now scientists are using small things, small robot, small machine that can also do our maximum work, that is nano robot. When they are injected in the patient, also they diagnosis and deliver the treatment, give the perfect treatment. Affected area can be repaired, and this nano robot get digested. And so such type of micro machines, that is the nano machines, can also be used for medical field. And now in case of future strategy, we can say that the new generation of scientists and workers skilled in a nanoscience and nanotechnology. So this is the area where the researcher can do much work as they like, and also new field, it is a new field, and also now new areas are there now also in this case also, and therefore we have to use this concept in mathematics, science, engineering, and in technological education, and scientists prepared work for the nanotechnology area. So there are a number of areas where we can work, and also we can give training to the society. Now it is expected that over next 10 to 20 years, nanotechnology will fundamentally transform science and technology and the society. So here, these are the summary of this. Number of methods we have seen, and also lot of applications are there of these materials. So it is a very new emerging field, and also having number of applications in the. So in short, also we can say that it is beneficial to the society, and there is a great impact of the as we know that nano car also came, and so nano materials used in the different magnetic materials, and day by day the technology going to be changed. And what is the our TV also is a very thin. What is now is going in very small size also. One scientist also says that one pen can contain by using the nano materials, we can see in that pen also there may be a whole world, the internet may be there, all the things we can capture in the small things. So one pen can cover all the whole world containing. This is the magic of these nano materials, and there is a technology, and so this is the usefulness of this material, can be used for different purposes in different field. This is very emerging field where we can work for the different applications. Thank you.
Thank you so much, sir. So, sir cover give the importance of a nanomaterial, that why, how, and where the nanomaterials are to be used, how the preparation of a nanomaterials, and how the we will change the particle size, how the property of the nanomaterial can be changed. Sir also cover the various methods and for the synthesis of a nanoparticles, such as Sol-Gel process, solver thermal process, hydrothermal process, etc. So I am very much thankful, sir, for this brief information on the nanoscience and its uses for the society. So, sir, there are some doubts from the participants. That the first doubt is from the Bahirwar sirs, that is whether there is an impact on the intellectual property issues.
Hello.
There is one doubt from the participant, that is from the Vaibhav sir, that whether there is an impact on the intellectual property issue.
Whenever we are discussing the intellectual property, we should have to take the care only. we can synthesize, we can do the method, but our property should be different. Application part is also different. Intellectual property, we cannot use other, we can see the method as used by the other scientists. So we can modify the methods. We can use the same method, but the properties can be different.
Another question again from the sir, that how the nanotechnology will have the impact in the military appliances, military applications.
Yes, sure. It is very beneficial for the military purposes. And now scientists are they have developed the material, such material that if you use this nano material and it is mixed in of fabrics, then the soldier maybe disappear. He can see the enemy, but he his cannot be visible. So in this way, he can attack like that our enemy. That the fabrics made by these nanomaterials are very useful for the soldiers and also in other field also. So it is very useful material. Yeah. Nano jacket can also be free.
One more question from the participant. Can you elaborate the use of nanomaterial for water treatment process?
Yes.
Can you elaborate?
Yes. Okay. As we have seen number of applications, and one of the application is to reduce the pollution, and also for the water treatment. So many different types of nanomaterials or nanoparticles are used in water treatment process, and nanotechnology is useful in regards to remediation, desalination, filtration, and also purification. And water treatment, for the water treatment, so we can say that the main feature that make nanoparticle effective for the water treatments are they are number of parts are there. So there are many water purifier available in the market which are used for the different techniques like boiling, filtration, desalination or distillation, chlorination, sedimentation, oxidation, and nanotechnology play an important role. That nanomembranes are used with the purpose of water softening and removal of contaminant such as physical, biological, and chemical. And it is because main feature we can say that is the water treatments are effective. It is effective because in the water treatments are they have the more surface area, small volume is there, higher surface area and volume, and therefore particle becomes stronger and more stable and durable. Material may change the properties and electrical, optical, physical, chemical, biological, and at nano scale, and therefore it is very much useful for the chemical biological reaction also. So we can say that this material or technology can be very beneficially or can be useful for the water treatment process. And scientists are using this process introducing the material as a absorbing materials and also for softening the water also. So it is much useful.
One more question from the paper, that what are the limitations associated with the nanoparticle toxicity?
This is an important I have discussed. There should be, we should have to take the care, and the toxicity is there because it is a nano size. We should not see that, and whenever we are working with this, we should have to wear the hand gloves and also mask and all the precaution we should have to take in the laboratory whenever we are in the dealing with the synthesis or any characterization part of this nanomaterial, because there are some environmental issues also there and also legal issues are also there, and we should have to take care, because if we inhale such type of small particles, so it can cause it is dangerous. So we should have to take, it is toxic, very toxic. So we should have to take the care when we deals with this such type of material, and therefore there should be some discussion international level or national level. We should have to take such type of discussion. We should have to take this some remedy about this. So this is that that is why I told also some precaution we should have to take.
Thank you so much, sir. You covered all the uses and limitations and all the that is the method of synthesis about the nanotechnology. I think we have to stop here, sir. Thank you so much. Again, I am from the Nano College of Science. I am heartily thankful to you, sir, for the sharing your deep knowledge with us. Thanks, sir. So, proceeding further, thank you very much for all the sirs and ma'am for sharing your deep knowledge with full of enthusiasm. Ladies and gentlemen, your presence made this day memorable. Thank you for listening, encouragement, and most importantly, giving your time and making your presence. So, with this, at the last part of the event, may I request convener of today's webinar, Dr. Khobragade ma'am, proceed further for the vote of thanks. Please, ma'am.
Yes, sir. I have expressed my of immense pleasure in having this honor of my deepest sense of gratitude to President, Resource Person, Principal, Coordinator, Organizing and Participant. This has been very nice of all the guests decided and enhanced the grace of the program. First, I would like to thank respected Honorable Sardar Naginder Singh Ji Soni, President of Guru Nanak Seva Samiti Viru. He always have been source of inspiration for us and kindly support in organizing the webinar. I express my sincere gratitude to Honorable Professor Gladys Completo from Philippines, who accepted our invitation and delivered a graceful speech on this occasion. We all have been enlightened and motivated by your talk. I am extremely thankful to Honorable Dr. Tofida Mohammad Kabir from Nigeria, who accepted our invitation and delivered an excellent and knowledgeful talk. I am personally and on behalf of organizing committee very heartily thankful to our resource person, Honorable Professor W.B. Gurnule, who delivered excellent talk. Sir, without your help, this webinar would not have been possible. I extend my thanks to our principal, Dr. B.M. Bahirwar, sir, principal of Guru Nanak College of Science, Ballarpur, for guiding me from time to time to make this webinar a grand success. I express my thankfulness to coordinator of today's function, Professor P.B. Ghode, sir, head of the Department of Chemistry, Guru Nanak College of Science. Sir showed the keen interest and kind advice with words of encouragement has made this webinar success. I am also thankful to all my colleagues, Dr. Y.G. Bodke, Professor S.D. Gedam, Dr. N. Jutode, Dr. D. Deshmukh, and Dr. N. Jadhav, as they came out with helping hands to put this webinar on success. All the staff members of our college also need to be thanked because they did not leave unturned to make this webinar a grand success. I am also thankful to all the participants for giving a warm response to the international webinar. At last, I give thanks to all those who have helped us directly or indirectly to make the webinar a grand success. Thank you.
Thank you, ma'am.
Thank you, sir.
Thank you, ma'am. Feedback link will be shared in the chat box, participants. So with this, I declare the completion of today's international webinar on the multidisciplinary research. Have a good day and good night.
Hello, madam.
Yes, sir.
Hello, all the participants on the Zoom can on their camera so that we can take the group photographs. Hello.
Okay, sir. Yes, sir.
The participants are requested to hello.
Yes, sir.
Yeah.
Thank you so much, sir. Gurnule, sir.
Thank you.
Thank you, sir. Gurnule, sir, thank you.
Sir, thank you. Okay.
Thank you, sir. Very well.
Good, sir.
Sir, Bhaiva sir, 2 minutes. Bhaiva sir, 2 minutes. सगळ्यांचे कॅमेरा ऑन करा सो दॅट वी कॅन टेक द फोटोग्राफ.
ओके सर. माझा बंद पडला.
हॅलो, पार्टिसिपंट आर रिक्वेस्टेड.
कॅमेरा ऑन करा. सर्वांचे कमेंट घेतो.
Thank you so much, sir.
हॅलो.
तुम्ही बसा आणि